Good morning, everyone. We are in the last day for our successful meeting. So it's my honor to introduce Dr. Michael Puraganan from the University of New York. He's going to talk about natural selection on plant gene expression. Go ahead, Michael. Welcome and thank you for accepting our invitation for this Congress.
Okay, buenos días a todos, mis colegas en Mexico. Thank you very much for this opportunity to present our work with you. Thank you, Ulysses Rosas, Professor Ulysses Rosas, for inviting me to give this seminar. I hope you can all hear me. I'm assuming that you can all hear me.
Yes.
Yeah.
Okay, good. And I am Filipino, and my mis padres hablaban español, but my Spanish is not very good. So for the rest of the seminar, I'm just going to speak in English. I'm sorry for that. So my laboratory for the last, in its entire history, was really focused on trying to understand the genetic basis of biodiversity. We're interested in trying to understand the mechanisms, both the evolutionary mechanisms and the genetic mechanisms that lead to the species differences we see across the world, and especially in plants, because I'm trained as a plant biologist. And since I was a graduate student, a lot of my efforts have actually been focused on looking at how gene regulation and gene expression has evolved. And there were many reasons for me to be interested in this. But one of the major, shall we say, one of the reasons that I became interested in this was reading this paper several years ago by Alan Wilson and Mary Claire King. And Alan Wilson, for those of you who are too young to know, is one of the founders of the field of molecular evolution. And Mary Claire King, somebody who discovered the breast cancer gene, and she's famous for that. But you might not know that she was actually trained as a molecular evolutionist. And they wrote this very important paper in the 70s, wherein they suggested that much of the species differences we see, the differences we see between humans and chimpanzees, for example, are not because of changes in the protein level of just the general proteins in the organism, but might be because of changes in regulation, because they didn't see many differences in protein differences between chimpanzees and humans, even though if you look at chimpanzees and humans, they're very different phenotypically. So since then, I really have been working on and off and trying to understand the evolution of the genes involved in regulation, as well as the evolution of gene expression and gene regulation itself. So when I was a graduate student, one of my thesis projects and one of the papers I published was looking at the evolution of the basic helix, loop helix plant regulatory genes, looking at those genes responsible for color pigmentation in various plant parts, and showing that, in fact, even though people thought that regulatory genes were strongly conserved in evolution, I actually showed that, in fact, large portions of regulatory genes, at least the regulatory proteins, are evolving very fast, contrary to what people thought. And then, during my postdoc, I also continued to look at the evolution of regulatory genes. This time, you might remember this. This was work I did as a postdoc looking at the evolution of the plant Madsbach genes. We, of course, know the plant Madsbach genes are those genes involved in flower development. And again, by looking at the phylogeny and the molecular evolution of these genes, we were able to learn something about how the flower development had evolved throughout the history of land plants. And we continued these with a variety of studies. This was a study that actually was done by Ulysses Rosas, who's now at UNAM as a professor. He did this as a postdoc in my lab, where he was able to demonstrate that mutations in the cis-regulatory region, changes in the copy number of a microsatellite in the cis-regulatory region of the Constance gene may be involved in variation in flowering time in Arabidopsis. And even as late as 2 years ago, we're back to color, where we have projects in date palms, and we showed that changes in the color of fruits in the date palm is associated with a mutation in another regulatory gene, the MIB regulatory gene. So as you can see throughout my career, much of the focus of our work has been on trying to understand how regulatory genes and how cis-regulatory regions have evolved. And this is a work that we're continuing in my lab. So today I'd like to talk to you about much more recent work we've been doing on looking at selection and gene expression, especially on plant gene expression. And in doing this project, really what we want to be doing is to look at two things. We want to look at the evolution of maybe the cis-regulatory regions, or trying to identify, using evolution to identify cis-regulatory regions in the plant genome, and also looking at gene expression or transcript levels in a plant, and treating this like any other phenotype and trying to understand how selection operates on transcript levels or in gene expression. So let's start off with looking at gene expression. Rice, and we work here on rice. Rice sativa, which is one of the most important domesticated species in the world. And this project that I'm going to talk to you about in the next few minutes was led by Niels Groen, a postdoc in my lab, who's now a faculty member in the University of California at Riverside. And this was published last year in Nature. And the idea behind this comes from trying to understand how selection operates on phenotypes. So we're very familiar with many classic examples of selection on adaptive evolution on phenotypes. So one of the famous examples, of course, are Darwin's finches, where the shape and sizes of the beaks are associated with episodes of selection in the Galapagos Islands. This was, of course, done by the Grants at Princeton, where they showed, for example, that the depth of the beaks of the birds shifts after a drought episode on the islands as a result of the change in the types of seeds that grow before and after a drought. So this shows an episode of selection that changes a phenotype here that we see in the Galapagos finches. And for the longest time, evolutionary biologists have been interested in trying to understand these examples of selection, not only to show that they exist, but to also quantify it. Like, how strong really is selection on these different phenotypes? And there have been several ways in which people are able to quantify or to figure out how strong selection is acting on various phenotypes. And the most important one comes from multivariate selection theory, which emerges from quantitative genetics. And in multivariate selection theory, the response of a phenotype to selection is just equal to the amount of genetic variance in that phenotype plus the strength of selection. Okay, so this is what we call the breeder's equation. Response to selection is a function of both how much genetic variation there is in a population and how strong is selection acting on that genetic variation. And what happened, nearly 40 years ago now, was that Landy and Arnold looked at the multivariate selection theory and basically came up with a way to try to measure the selection gradients or the strengths of selection on various phenotypes. And the way they do it is says they look at fitness of an organism, and they say fitness is correlated to the phenotype in a way that is correlated to the strength of selection, both linear selection and what's called quadratic selection. So there's a relationship between phenotype and fitness. And by looking at the relationship between phenotype and fitness, you can measure the strength of what's called linear selection and quadratic selection. And in their formula here, if you get beta, this term here, which is called the selection gradient, the linear selection gradient, if there is a positive or negative correlation between fitness and a phenotype, that is beta is greater than 0, then we say that the trait Z is under directional selection or under positive or negative selection, but under directional selection. Also, if this term gamma is not equal to 0, you can see other forms of selection, say stabilizing or disruptive selection. Stabilizing selection happens when gamma is less than 0, so that in this type of selection, intermediate phenotypes have the highest fitness, while extreme phenotypes have lower fitness. And disruptive selection is gamma greater than 0. So when gamma is greater than 0, then the intermediate phenotype actually is the lowest fitness. It's the extremes that have strong fitness. So basically what Landy and Arnold said is, if you are able to measure the fitness of an organism and their trait phenotype, you can actually estimate the strength of selection, either the strength of directional selection or stabilizing and disruptive selection. And because of this, actually, since this paper was published in 1983, there have been thousands of studies that have measured the strength of selection in a variety of different organisms and a variety of different traits. So really now there are probably more than 1,000 measures of selection and a variety of phenotypes. And these databases of strength of selection are so large now that we can begin to look at the any patterns on selection that we see both in the wild. So for example, this is an old review now. This is 20 years old, but the pattern here is the same. What we can see is that linear selection, which is directional selection, so here fitness is directly correlated with the phenotype. The strength of selection looks like it's a long tail distribution. So most of selection is actually relatively weak. Selection gradient is relatively small, but there's a long tail to the distribution. So there are some, it's not a lot, but there are some selective forces that are actually fairly strong. And we can see how that's different for things like morphology. And this is, by the way, in animals, how the distribution of the strength of selection for morphology versus things like life history, and what the differences are. So you can see how these types of analysis can be fairly powerful. It can tell us something about the strength of selection across organisms and how these may be different in different classes of phenotypes or different types of traits that we see in the wild. So I had this idea several years ago that maybe we can measure the strength of selection not on organismal phenotypes, such as flowering time or the size of the plant or the height of the plant and so on, but maybe we can measure the strength of selection on gene expression. That is, look at transcript levels in the plant and treat that as a phenotype and see how strong is selection on gene expression, on transcript levels. Now, in order to do that, you can see what we need, right? We need to find some way to measure the levels of transcript in a plant and also measure the fitness, say the number of seed. And for a long time, this experiment could be done, but it was very expensive to do. But with advances in genome sequencing technology, it became possible to do this. And so several years ago, about 5 or 6 years ago, we decided, let's try to do this. Let's try to do this in a genome-wide fashion in a plant. And we decided to do this in rice. So what we decided to do was to do an experiment where we planted 200 land races or traditional varieties of rice in the field, both under normal conditions, that is under wet paddy conditions, and under drought stress conditions, so under a condition of an environmental stress. And by growing 200 land races in the plant, we're able to do several things. First, we're able to phenotype. We're able to phenotype various phenotypes, both under normal conditions and under drought stress. We're able to do things like carbon isotope analysis, chlorophyll content, the osmotic potential, the architecture of the roots and shoots, flowering time. But there were several important things for this particular experiment that we wanted to measure. First, we wanted to measure levels of transcript. Here we did it in leaves. And we did this including replicates for the 200 plus land races plus replicates. And in the two conditions, we essentially looked at RNA levels across the entire genome for 1,320 samples. So this was a very large scale experiment. And then we also wanted to measure fitness. And here we measured both grain number, and actually I talk about mortality, but it's not mortality. We basically whether the plant flowered or not. So it's flowering success. So we look at grain number especially and flowering success as our measure of fitness, and we measure the levels of RNA or transcript levels. In order to measure transcript levels at a very large scale, as you can imagine, doing it for 1,300 samples could be very expensive and could take a long time. But what we did is we modified a protocol developed by colleagues here at NYU and at the New York Genome Center, where this was a method that was used for single cell transcriptomics. And what we did was modify this to work in just normal RNA-seq, where you can essentially attach a transcriptome to microparticles. And then by doing that, you can create molecular tags that can tag each transcriptome to particular samples that we did. And so we were able to modify this. And by doing this, we can create 1,000 libraries in a matter of 1 week or 2 weeks at the most. So we were able to create the libraries very, very quickly and very inexpensively with this method. So with this, we were able to then assay transcript levels and expression of about 15,000 genes across essentially, 1,300 samples. That is about 220 varieties in 2 environments in replicated conditions. So we're able, and this was looking at all heritable transcript. Of course, you can't see anything here, just to show you that we're able to measure the expression level in a very large number of samples across a large number of genes in the genome. And the other thing we were able to do with this experiment is our colleagues at the International Rice Research Institute in the Philippines was able to measure fitness. And in doing that, we measured many aspects of fitness, but the two that was important was the number of seed that was produced. So some individuals would produce a low number of seed, some would produce a large number of seed, and we were able to count this across these 1,300 samples. They essentially counted over 4 million seed, both using automated procedures and in some cases by hand counting. And so we have a measure of fitness here in this equation, and we have a measure of gene expression. And so by applying this method by Landy and Arnold, if we have a measure of fitness and we have a measure of our phenotype, this is gene expression, we can measure the strength of selection. And one of the things we had to do here was, in fact, the problem here is usually when you do Landy and Arnold, you do this in a multivariate framework. That is, you put all of the phenotypes in one model. But of course, there's 15,000 traits here, 15,000 gene expression levels for genes. And so we couldn't do all of those simultaneously. So we did this one at a time. For every gene, we created its own model for selection. And in the end, what we did was we were able to get a distribution of the strength of selection under both normal conditions and under drought stress in rice. And this shows you some of the pattern here. First of all, just like you would see in organismal phenotypes that others had shown, most of the strength of selection is actually fairly close to 0. So what we're seeing is that on gene expression, the strength of selection is largely neutral or it's very close to neutral. However, there's also expression levels that are that for some genes that are actually fairly strong. There are not a lot of them, but there are examples of genes in which the level of expression is experiencing very strong linear selection. The other thing that we notice is that under drought stress, what one sees is that the strength of selection actually shifts. Under drought stress, selection actually becomes stronger for gene expression. It shifted to, in fact, it shifted to more positive levels. So the strength of selection shifts under drought stress, so that under drought, gene expression the expression of individual genes experience a stronger selection. And this is how you can see the shift also when you look at absolute levels of selection. That is, we look at both negative and positive selection, but we look only at the strength of selection. You can see the stronger shift of selection under drought stress compared to normal. But the exciting thing here now is we can now measure the strength of selection, of natural selection on the expression of individual genes. So before we can measure the strength of selection on phenotypes like height or weight or flowering time and so on, now we can measure it on a molecular phenotype. This is the level of gene expression. And hold on. For some reason, my slides are not advancing. Let's see. Okay. And as I said, I already mentioned this. If you look at the distribution of the strength of selection, it's very similar to what we see in organismal phenotypes that have been seen by others elsewhere. So qualitatively, what we see in a molecular phenotype, such as gene expression, is very similar to what we see in organismal phenotypes, such as morphology or life history. We were able to also see if there are different types of factors that affect the strength of selection on gene expression. I show you one example here, that connectivity of a gene. So, of course, genes do not actually act alone in the organism. They interact with other genes. They form networks of gene interactions. And what we did was, for our genes in our analysis, we looked at the connectivity of those genes. Were they connected to a large number of genes, that is, they had high connectivity, or a low number of genes, that they had low connectivity? And what you can see here is the strength of selection appears to be higher if the connectivity of the gene is low. If the connectivity of the gene is high, the strength of selection is lower. And the reason for that, we think, is because genes that are highly connected in networks will have very pleiotropic effects if you change their gene expression. That is, they affect a large number of other genes. And so selection wants, basically, you don't want to have this gene, you don't want this gene to change its expression very much. While genes that have low connectivity, maybe their effects are much more targeted, and so selection can act on those genes much more readily. And we basically looked at different types of factors that may affect this. So, for example, how stochastic is the expression? What's the level of expression? So, for example, what we find is that genes that have higher levels of expression have lower selection strengths, the housekeeping genes, compared to genes which have lower levels of expression, because they might be inducible genes or things like transcription factor genes and so on. So we're able to look at how different factors of the genes themselves affect the strength of selection on gene expression. We're able to also show what kind of genes are affected, what groups of genes are affected, or what the strength of selection are on different groups of genes. So under normal conditions, what we see is that under normal conditions, we see a variety of groups of genes that are affected. Many of these are in things like response to herbivory, response to pathogens, so for example, defense response. Under normal conditions, expression of genes seem to be under stronger selection if they're involved in things like response to pathogens. While under drought stress conditions, what we see is we see stronger selection for genes that are involved in drought response. So for example, ABA signaling, but also in general in growth response. So things like leaf development or regulation of growth are under stronger selection. Under drought response, actually, plants want to essentially shut down their metabolism. And so genes involved in the regulation of growth become much more important. So we can see a shift in which genes have stronger or weaker selection under stress conditions versus normal conditions. And we can look at individual genes. For example, under drought conditions, what we see is that genes involved in photosynthesis, these individual genes, and genes in particular, like the rubisco small subunit and the rubisco activase, under drought, have very strong selection strengths. In fact, what happens is that under drought conditions, these two genes involved in photosynthesis appear to be selected for increased expression. And in fact, right now, what we're doing is we're creating transgenic lines in rice to see if by increasing the expression of these genes in rice, we can, in fact, help make higher yields under drought stress. One gene in particular that we were interested in is this gene, Madsbach's gene 18. This is a gene which we showed that under drought stress, it has actually one of the strongest strengths of selection under drought stress. The strength of selection was 0.77. You can see the p-value is 10 to the minus 11. What we saw in our study was this gene, the higher the level of expression of this gene, the higher the level of the number of seed the plant produced under drought stress condition. And what we realized is what this gene appeared to be doing was it appeared to be involved in flowering time. So when you increase expression of this gene, it leads to early flowering. And in fact, there's a major QTL for flowering time in this gene in rice. It explains 65% of the phenotypic variation in flowering time, a QTL that involves this gene. And it had already been shown, in fact, that a QTL for drought stress, for fitness under drought stress, was actually on top of this gene as well. So what we think this gene is, it's a drought escape gene. So when you are under drought, plants that are able to flower earlier are able to have higher fitness. And what this gene does is if it's got higher expression levels under drought, it promotes early flowering and leads to drought escape for the plant itself. And so again, we're also creating transgenic lines of this plant to see whether we can test this hypothesis directly. But you can see what happens in these types of analysis. They can be very powerful because what they do is that they're able to link expression of the gene to fitness directly and to measure how strong that relationship of expression of this gene is to fitness. And not only does it tell us something about the evolution of gene expression in this plant, for example, Rhiza sativa, or gene expression in organisms in general, but in this particular case, it also helps us help breeders who are interested, for example, in drought stress by giving them examples of genes that they may want to concentrate their attention on if they want to develop new varieties of rice, for example, that may help under drought stress. Oh, by the way, and this last slide here just shows you what I just said about flowering time, that in fact what you see is that the fitness of the plant increases on early flowering and decreases with late flowering. So I've shown you some data where we've looked at the expression of genes and treated the expression of genes as a phenotype. And in so doing, measure the strength of selection on gene expression. And if you want to read more about it, as I said, it was published in Nature last year. Now I'm going to switch gears a little bit and show you how we can look at evolutionary genetics and combine it with functional genomics in order to look at how selection has shaped the evolution of different parts of the genome and help identify especially genomes, regulatory elements in the plant genome, those in non-coding regions where we might not have good functional genomic information. So this is a project that we call the Rice Fitness Consequence Map project that was led by Zoe Jolie Lopez, another postdoc in my lab. This was published last year in Nature Plants. Zoe is now a faculty member at the University of Quebec back in Canada. And this project started again about 5 years ago as a way to, in a sense, a way to identify regulatory regions in the plant genome by using evolutionary genetics. And the idea here is the following, and this is from work by Adam Sipple at Cold Spring Harbor Laboratories. If you look at a genome and you look at different functional genomic measures, for example, open chromatin, like DNA's hypersensitive sites, what you see is that in the genome, we usually are able to annotate the protein coding region very well. However, non-coding regions are difficult to annotate because the amount of functional information we have within non-coding regions is very sparse. There are now, of course, many ways to annotate non-coding regions. For example, DNA has hypersensitivity. But even though we see epigenomic or functional genomic marks in non-coding regions, we still don't know if these regions are indeed functional. That is, there's a functional consequence of these regions to the plant or to the organism. So what Adam Sipple did was develop a method based on population genetics called the fitness consequence score. And what he did was he said, well, if you look at mutations in the genome or polymorphisms, we know that polymorphisms have different impacts on the fitness of an organism. Many mutations that you see are polymorphisms you might see in an organism are actually lethal or very, very deleterious. So these are very bad for you. And in fact, we never see these mutations in as polymorphisms in the genome because they are selected out really quickly. However, many polymorphisms that we see in the genome are there because they're either neutral or they're just slightly deleterious or they might be advantageous. Okay, so what we see as polymorphisms in the genome are usually mutations that are either neutral or nearly neutral. They're either slightly deleterious or slightly advantageous. Those mutations that are lethal, we don't see because they are selected out. Those mutations that are very advantageous, we see as fixed differences between populations or between species, because mutations that are highly advantageous are swept into high frequency in the population and become fixed really quickly because they're advantageous. And by using the spectrum of mutations in the genome, he said, we can then estimate a score for the impact of mutations in a region of the genome. And he called these the fitness consequence score. So for example, this region of the genome in humans, which had open chromatin because it had DNAS1 hypersensitive sites, also showed high fitness consequence scores. That is, mutations in this region had a high high probability of having an effect on fitness. That is, this region of the genome was functional. And in order to calculate this fitness consequence score, basically you use principles of evolutionary genetics. And the way you do this is using a method he developed called InSight, which is really a population genetic method where you look at polymorphisms in the genome versus fixed differences between the species you're looking at versus another species. And by looking at the amount of variation within the genome and the amount of evolution between that species and another species, you're able to calculate a variety of different selection parameters across the genome. And the one in particular we're interested in is this number rho. Rho here is the fraction of sites in a region of the genome that is under selection. That is, rho, rho tells you the strength of selection that is acting on a particular region of the genome. And as I said, in order to do this in rice, what we had to do is we had to look at both data between species, and here we look at 12 Oryza species, that is Oryza sativa, the rice that we eat, versus wild rice. And we were looking more specifically at Oryza sativa. So we had about 1,400 to 1,500 individuals where we had population genetic data. We had sequenced, we and others had sequenced the genomes of 1,477 individuals. So we knew the polymorphisms within Oryza sativa. So we adapted this method by Adam Sipple at Cold Spring Harbor called InSight, and we developed what was called Green Insight in cultivated rice. And in doing that, we were able to assign fitness consequence scores to different parts of the rice genome. Now, the way fitness consequence scores work is if you get a score of 0, score of 0 means essentially there's no selection in that region. That region is behaving neutrally. A score of 1 means that region is under very strong selection, so that mutating any nucleotide in that region has an effect on fitness in the organism. And as I said, you're able to estimate the fitness selection score based on population genetic and molecular evolution data. And here on rice, based on our data, we're able to see neutral regions have a fitness consequence score less than 0. Conserved non-coding sequences have a very high fitness consequence score. Coding regions have a distribution, but a lot of them have high fitness consequence scores and so on. So we're able to do it for very large types of classes in the genome: promoters, 5' UTRs, introns, coding regions, 3' UTRs, and so on. The question is, can we do this at a higher resolution? And Adam suggested a way to do it in a higher resolution. He said that if we have a lot of functional genomic and epigenomic marks in the genome, we might be able to classify different parts of the genome based on a combination of these different epigenomic and genomic marks. So instead of saying that a region is a non-coding region, that is, the class is non-coding region, we may be able to further subdivide that non-coding region in different subclasses based on the types of epigenomic and genomic marks that that region of the genome has. And so he developed a method that was able to look at the fitness consequence scores. And this method is in 4 steps. The first is to acquire a large number of genomic and epigenomic signals across the genome. The next step is then to use those epigenomic and genomic marks to classify different parts of the genome based on different combinations of these marks. Then to apply this insight method on those combinations of epigenomic marks, looking at the levels of variation and differentiation in the genome sequence level. And then by using that, you're able to classify or to estimate rho, which is the fitness consequence score. So let's look at that in rice really quickly. We're able to identify functional genomic data. So Zoe was able to get the levels of RNA sequencing, looking at levels of RNA transcript and small RNA transcript across the genome. She used ATAC-seq to look at chromatin accessibility. She had DNA methylation levels. She was able to get 6 histone marks in the genome, of which only 4 are actually important. The other 2 are actually correlated with the others. So only 4 are independent histone marks. And she was also able to look at RNA polymerase activity in the genome, whether there was bound RNA polymerase that was doing active transcription using a method called precision run-on sequencing. This is actually a fairly new method. It's a fairly difficult method, but she was able to work on it and use it on rice. And in so doing, she was able to look at the rice genome, and she had all of these genomic and epigenomic marks. She was able to acquire histone marks, methylation, small RNA-seq, active polymerase, open chromatin, and so on. So with that, using that data, she's able to aggregate regions of the genome that appear to have the same combination of these different marks. So for example, you can do this by eye. So this is, for example, a region that has open chromatin. It looks like it's near a region of that's being transcribed. There is active polymerase and so on. So you can look at it visually and see this is one combination of marks. But there's a way to do that using more sophisticated methods like hidden Markov model methods and looking at the annotation of the genome. Essentially, at the bottom line with looking at hidden Markov models and functional annotation, she was able to identify 246 genome classes in the genome that were important in the combination of different epigenomic marks. And these were distributed across the genome. Many of them were really small classes. That is, the regions of the genome were fairly small. Some were a little bit larger. But she was able to classify the rice genome as whether they belong to one class of chromatin marks or other classes of chromatin marks. And so with that, she was able to then use the insight method to estimate rho across the genome, and then to map that on the genome. And so in the end, what you see is you've got, a region of the genome or the genome of rice, and you're able to assign a fitness consequence score. For example, this is a rice gene in the rice genome. You see that upstream of it, the fitness consequence score is low, but within the coding sequence, within the exons, the fitness consequence score is high. This is a region of the intron, for example, where the fitness consequence score is high, suggesting that this region of the intron of this gene has some regulatory properties that may be important for the expression of this gene. So, and we can do that across the entire genome, both across all genes and the intergenic region. And you can see how the genome behaves in terms of selection based on row across the entire rice genome versus humans. We're able to look at the distribution of row values and how they're distributed across that. We're able to see things like how is row changing in genes that are transcription factor genes versus other types of genes or genes that are involved in tissue-specific expression. What we see actually is that upstream of transcription factors and genes that are involved in tissue-specific expression, we actually see relatively high row levels for those regions of the genome. So promoter regions of transcription factor genes and promoter regions of genes involved in tissue-specific expression. And so you can see how powerful this is now. We can start to look at upstream regions and non-coding regions of the genome and see if there are any regulatory elements there based on essentially what we know about selection. So all of this now is in a database that we've developed, the RiceFitCons-MAP database. You can look at our paper in Nature Plants last year where it shows you how to get access to this database. For rice, we also have one for Arabidopsis as well, although the Arabidopsis one is not as high resolution as the rice fitness consequence map. And with these, we hope not only to understand how selection has acted on different components of the rice genome or the plant genome, but also hopefully it will be useful for plant geneticists, wherein if they have a non-coding region of the genome that they're interested in because they suspect that there are regulatory elements in that genome, they can look at the the Rice Fitness Cons map database and see which part of that non-coding region may be under strong selection and therefore may be evidence of a functional importance of that region. So to summarize what I told you today, I think that the study of the evolution of regulatory genes and regulatory elements provide insights into molecular evolution. The evolution of organisms and plants, including their morphology, their life history, but evolution in general. I've showed you two specific studies where now we can measure the strength of selection on gene expression itself to gain an understanding of what processes are evolving across the different types of genes in the organism and across different environments. And also I've shown you a method by which when you can combine functional genomics and population genomics, you can help identify what we think are regulatory elements in the genome, especially in non-coding regions of the genome. So as I said, all of this, trying to understand the evolution of regulation in genes is to help us understand how diversity has evolved in plants. And with that, I'd like to thank all of these people who have helped in this project. As I said, the project that I talked to you in particular was by Niels Groen and Zoe Julie Lopez, and these are collaborators on that project. And with that, I thank you for your attention. I went up a little bit over time. I'm sorry for that, but I hope there's time for at least one question. Thank you.
Thank you very much, Michael. You have a very, very nice data and a wonderful talk. So we have time for one short question, please. Yeah, there is a question from Alejandra. Go ahead, Alejandra.
Yeah. Oh, thank you for your great talk. I really enjoy it. I am curious whether you think that if you think that the same fitness trend would be if you use different genotypes or if you use different drought treatments, like let's say like a reproductive terminal drought, for example.
Right. Thank you for that. We don't know. So we've actually done this experiment also in salinity. And so we'll see if different environments, all the salinity is related to drought, if that changes the dynamics of selection on expression. We're also interested in what's happening on gene expression, like you said, for example, in the reproductive organs. The problem is it's hard to stage the tissue, the collection of tissue, so that they're the same stage across all different rice genotypes. So there's a logistical problem with that. But I suspect, in fact, we might see something very different if we look at reproductive tissue, and that would be very interesting.
Yeah.
As to populations, we looked at here Oryza sativa itself as an entire population. And that's another question which we would like to do. What if we look at another Oryza species, maybe Oryza glaberrima, African rice, or wild Oryza species? Of course, the selection, we would be interested to see what groups of genes have similar patterns of selection and which groups of genes have different patterns of selection. And I think that's a good area to explore in the future.
Yeah. Okay. Yeah. Thank you.
Yeah,
thank you very much. Again, Michael, there are some comments in the chat saying that your work is very, very, very nice and excellent. And there is another question in the chat, but we don't have time. Please, if you can follow it in the chat. Thank you very much.
Thank you.
Our next speaker is Tatiana Arias from the Tecnológico de Antioquia, Medellín, Colombia. She's going to talk about towards a solid orchid phylogeny, providing a solid time frame of biography and macroevolution studies. Go ahead, Tatiana, please. Share your screen.
Okay, so can I start now?
Yeah.
Okay. Well, actually, I want to start saying that I don't work at the Tecnológico de Antioquia anymore. I am going to move to the Mary Selby Botanical Gardens in Sarasota, Florida next week. So you all know. So good morning, everyone, and thank you so much for being here today. In particularly, I'd like to thank Dan Chitwood for inviting me to participate in this conference. I feel very, very honored by this. Today, I want to talk to you about the research I have been doing for the past few years in orchids and the new perspectives that we have about this work. Let's see. So the first question that I would like to answer is why we should bother about studying them. What is so special about orchids? And orchids are herbaceous plants that have terrestrial or epiphyte, and they have a spectacular number of species worldwide and also genera that grow pretty much in every environment you can think of. They have also a great deal of floral diversity and also have developed interactions vital for the completion of all their life cycles with other organisms such fungi and animals, and in particular insects. Orchids are super diverse in the northern Andes, particularly in Colombia, where the Andes divide in 3 mountain ranges. And they are, for this reason, because they are super diverse and are in all these ecological geographical regions in Colombia, they are very fascinating and a very good model organism to study evolution and the diversification of monocots in mountainous environments. So despite being so diverse, orchids have a very modest number of molecular resources available, for example, in gene bank. And a quick search that I just did yesterday shows that there are 70 nuclear genomes available for the orchids, and most of them are within genera like Dendrobium and Phalaenopsis that are important horticulturally and that are also Asian genera. There are also some genomes available for vanilla, species that is highly cultivated in Mexico and very important, economically important there. And you can also see that there are some complete plastic genomes and some other molecular data or genomic data that could be mined, that could be potentially mined to better and understand molecular processes and mechanisms within the family. So I'm going to talk about phylogenies, right? And in 2000, I want to give you first like a historical point of view of what has been going on in this field. And in 2015, the first phylogenomic study was published, the first phylogenomic study for orchids, of course, was published by Givnish and collaborators. And this study included 75 coding regions of the chloroplast, so a big deal of information, and about 39 species representing the main subfamilies and some of the tribes and subtribes within the family. Okay, so you can see color-coded here, the main subfamilies within the family. The tribes are here, subtribes here, and genera. And as I told you, there are more than 700 genera within the family, so the taxonomical sampling for this was still really low at this point, and is still really low. So besides being the first attempt to make a phylogeny using a great deal of molecular information, these authors also estimate diversification rates, diversification times, and talk a little bit about the evolution of the main orchid synapomorphisms. Of course, being this a really, or the first attempt to understand the evolution of the family at a genomic level, at a phylogenomic level, the taxonomic sampling was very low. And this is a conference of molecular evolution and biochemistry, so I don't know how familiar many of the people here are with phylogenetics. So perhaps some of you perhaps are not familiar with this, but the chloroplast is used very frequently in plants for phylogenetic reconstruction for many convenient reasons. Plastic are usually uniparentally inherited. They have a high number of genes that can be used to reconstruct phylogenies and also intergenic regions. And they are good markers in terms of tempo to resolve macroevolutionary questions among species and genera. So when you are going to do phylogeny, ideally you will want to have an uniparentally inherited markers like the one in the chloroplast for plants, but also biparentally inherited markers as the inherited in the nuclear genome. So early this year, my student Alejandra Serna with Oscar Perez, Diego Bogarin, and other, and also a group of other scientists, mostly from Latin America, we published an expanded orchid phylogeny, still using coding regions from the chloroplast, but including many more taxa at different taxonomical levels. And I think that we are seeing, and we are going to continue seeing this type of phylogenies for the orchid family. And this is going to be every time with a bigger taxon sampling that will ultimately give us a better picture of the mode and tempo of evolution in the Orchidaceae. So expect a lot of publications in the coming years about this. And again, because many of you are not familiar with this, there are many approaches to the way a whole chloroplast phylogeny can be built. But perhaps the most talked about method is the one known as genome skimming or genome survey. And in this method, this method is a way to superficially sequence a plant genome for very cheap. It's a cheap method to do this. And what this method do is take advantage of regions that are highly repeated in a total plant DNA sample. And if we think in a plant cell, there are hundreds of plus mitochondria, rDNA, and other housekeeping genes from the nuclear genome that are abundant. And because of this, they probably will have a higher depth of coverage during the sequencing that regions of the nuclear genome, for example, that are single copy. So once this sequencing is done, and as I say before, it can be done by this method or by other methods. But once the sequencing is done, in order to produce phylogenetic trees.
Pause.
Hello?
Just.
Okay. Once the sequencing is done, and in order to produce phylogenetic trees, we use bioinformatic pipelines like these. I mean, every person using phylogenomics is going to use a different program, and different programs are coming out every day. Some of them perform better for certain reasons, and etc. But the ultimate goal of this bioinformatic pipeline is to recover an alignment or a hypothesis of evolution in a phylogeny to do downstream analysis about or related to diversification or rates of evolution, etc. So, and this is how much all of the phylogenomic studies that use the chloroplast are done today. And in our paper, the one that we published early this year, we sequenced 78 plastin coding genes. We included 51 new species, and we included also everything that we could find out there related to the 70 plastic coding genes in the Orchidaceae. And for some of these species, we also were able to recover the whole chloroplast to study the molecular biology and molecular, or to describe the molecular biology of this chloroplast. And we focus mostly on tribes Cymbidium and Epidendrum. Okay. And I don't want to go in detail about this phylogeny because I know that you can barely see it. But I want to tell you that the chloroplast seem to be quite informative to resolve this phylogeny with good statistical support, both for most of the branches, as you see here, or if you can see that, for most branches. But still some nodes that are represented here by these red circles are difficult or are poorly solved here. And they were also poorly solved in the 2015 paper. And this can be due to several things. First of all, this can be due to poor taxonomic sampling again, that we still have at the genera and at the species level, but also it could be that the chloroplast did not provide sufficient information to resolve these relationships. And then in that way, nuclear markers need to be explored. And this is a way more difficult phylogeny for you to see here, but I just want to point out again that the majority of the unsupported nodes that you see here in red again are from the subfamily Epidendroideae, which is the most species-rich subfamily, and also the subfamily Orchidioideae. And when we go and see the sequences in these two subfamilies, we see that there is a low level of sequence variation within or in these particular relationships that are not resolved. But not all was that bad in this phylogeny. First, we were able to include way more genera in several subtribes, and also we were able to resolve some recalcitrant relationships that single gene or few genes phylogeny were unable to resolve, and that the phylogeny of Givnish in 2005 wasn't able to resolve to. And this is a case in the Cymbidium and in the subfamily Orchidioideae. This particular case that I am not gonna like detail. I mean, if you are an orchid biologist, you will be really interested in this, but not. And if you are not, probably not. So let's see why this is not working. Okay. Results from this phylogeny also allows to keep thinking about the evolution of highly diverse groups like the Pleurotalinidae. The Pleurotalinidae is a high or a rapidly diversifying subtribe that has approximately 5,000 species, and according to our diversification times, and other phylogenists too agree with us, has speciated, or this amount of species have been produced in the last 20 million years. And based on certain phylogenies or certain reconstructions that you have, there are two hypotheses out there about the evolution or about the ancestors that gave rise to the whole subtribe. One of these hypotheses says that ancestors come from the Antilles, and the other one said that they come from Brazil, and they enter or they're migrating to the Andes, and from there, the majority of the species, around 80% of the species, diversify between 16 and 5 million years ago. However, as I told you a little bit ago, this depends on the topology that is recovered in the amount of data, and particularly the amount of taxonomy sampling that we have here. So I think that we have to wait and see if this is one of these hypotheses is supported by a more complete phylogeny, in particularly taxonomically more complete phylogeny. So I want to make sure that the big take-home message of this talk is that orchids are still a group that is largely undersampled in terms of species and genera. And it's hard to make solid conclusions about the processes this way. But hopefully this will start changing soon. So besides phylogenetic trees and evolutionary hypotheses, these techniques also allows to recover complete chloroplasts, to first describe or to make a molecular description of these different chloroplasts, their sizes, the percentage of GC, etc., but also to understand the molecular evolution of these plastids across the orchid taxa. And in our case, something that has been discussed before, but that we also saw in the chloroplast that we were able to recover whole, we see that early divergent orchid species such as Apostasia, Vanilla, they have a great deal of NDH copies, while the plants that are epiphytes and diversified more recently have way less number of NDH copies. The NDH losses have been proposed by many authors to be associated with the tendency of orchids to grow in low-light canopy habitats. And they are related also to, that are related to epiphytism. The NDH complex function as an electron recycling system, allowing the fine-tuning of photosynthesis. And in dark environment, this might not be so essential. So it will be really interesting to keep exploring why some of the epiphytes, some of the orchid epiphytes have such a low number of NDH genes. So let's switch gears a little bit and talk a bit about the phylogenomic studies that have used nuclear data. And in 2009, and as a part of a really ambitious project that is called the Tree of Life, a set of nuclear probes known as the 353 set of probes, and this is referring to the number of genes that are recovered for flowering plants, was published. This set of baits was of probes was published, and they are mostly low nuclear copy markers that have been shown to work really well for phylogenetic studies across the flowering plants. And of course, Oscar Perez, as being part of Q and an orchideologist, have to try this set of probes across the orchids. So also this year, he published this first approach to this data set comparing the nuclear and the chloroplast phylogenies and using a big amount of markers. This data set, as you see here, have a pretty good representation for tribes and subtribes, but again, the taxa representativeness in terms of genera is really poor. So in this network, you can see that again, he was able to recover 6 different main groups that are or that coincide with the main subfamilies within the family, and yeah. But then next year, he is hoping to publish a big phylogeny, including more than half of the orchid genera that Oscar and other collaborators already sequenced using the 353 probe set. So the second thing that Oscar is showing in his paper is that when we go and look at the congruency among phylogenies, and I want to tell you here, this is the plastic data set that we produce, and this is the nuclear data set that Oscar and collaborators produce. And then we have here phylogenies at the genera, at the tribe and subtribe levels. You are gonna see that at every single taxonomical scale, you're gonna see that we have some incongruences among the topologies. And again, these incongruences can be the result of poorly taxon sampling phylogeny or a poor taxon sample phylogeny, or could also be an indication of some evolutionary processes such hybridization, horizontal gene transfer, among several others. So as conclusion for this slide, it seems that these nuclear markers are way more informative than the chloroplast, but I think that both approaches should be used when possible. So over the last part of my talk, and as a part of my new position as a research botanist at the Selby Botanical Gardens in Sarasota, I am going to tell you a little bit more about some of the orchid groups I will be studying in my new job. So I am interested in keep studying Andean orchids and the evolution of the epiphytism from a developmental and from a phylogenetic perspective. And I like to use orchids as model organisms to study ecological and evolutionary processes, particularly in northern South America. And for this, I will be working mostly at the general level in lineages that are super diverse, such as this genera in the Pleurotalini subtribe called Lepanthes. Lepanthes is a genera of more than 300 species that have really small size plants and really tiny flowers. And for this, I will be working with Laura Esseman. Laura Esseman belongs to the Atlanta Botanical Garden. And this year too, she published this set of baits that seem to be way more informative than the 353 set of props that Oscar used, and this mostly at the genera and species level. So in this study, I have formed a collaboration too with many different taxonomies of Lepanthes, such Sebastián Moreno, Sebastián Vieira, and Eugenio Restrepo. And I am also, and I hope to keep working with several Colombian universities, and in particular Alejandro Zuluaga from Universidad del Valle. So Lepanthes' richness and endemism in the northern Andes is incredible. And it seems the genera and the allied genera that are more than 1,500 species have diversified over the last 8 million years. And it seems that they are related, or the diversification is related to either mountain uplifts in the Andes, in the extreme mountain regions that we have in Colombia, or also has been proposed that this is due to pollination syndromes. So Sebastián Moreno, which is a master student from Univalle in Colombia, is studying niche modeling and niche and distribution modeling in the genus. And this analysis that I'm showing here show the particular areas where there are more species richness for Lepanthes, and also the high amount of endemism. This could be used in a phylogenetic context to understand the evolutionary patterns of biogeographical patterns and distribution patterns that are shaping these patterns, these phenomena or these distributions that we see here. And also this information is required for conservation in Colombia. So I know that he's using this to do conservation studies and to include some of these Lepanthes species in the red listing of the IUCN. So what we have done so far is that we did this big, using these areas of high diversity and endemism, we just finished a big expedition. Sorry?
2 minutes left.
Okay, we just went through we did this expedition in which we tried to collect the most amount of species across Colombia. So as conclusion of my talk, we provide an expanded, robust temporal phylogenomic framework of the Orchidaceae. This is a preliminary work for biogeographical and macroevolutionary studies. However, taxon sampling is still a big issue in the Orchidaceae. And nuclear versus chloroplast markers are still showing inconclusive or incongruent relationships for several taxa. So with this, I like to thank many people for all this is the effort of many people, students and collaborators, as I say. Lots of grant agencies have supported our work. And I have been recently appointed as recruiting editor for the journal Applications in Plant Sciences from the Botanical Society of America, and I am inviting authors to come and publish their work with us related to methodology or applications in plant sciences. And with that, I take questions.
Thank you very much, Tatiana. Unfortunately, we don't have time now for questions, but there is one question in the chat that you can ask directly there. I'm going to appreciate very much it. Thank you very much again.
Thank you.
Okay, so we go to the next speaker. Our next speaker today is Oscar Perez Escobar. He's coming from the Kew Royal Botanical Gardens from the UK. And he will be telling us about delving into the evolution of the date palm through aDNA and molecular clocks. Thank you, Oscar, for being here, and go ahead. The audience is yours.
Good afternoon, everyone. Can you hear me well?
Yes, yes.
Awesome. Well, thank you very much for inviting me to participate in this symposium. It is a great pleasure. And today I'd like to share with you the outcome of a project that my colleagues and I have been working for the last 3 years or so about the evolutionary history of the date palm as investigated through ancient DNA and molecular clocks. But before I start, I would like to clarify, to warn you that I am not a palm expert nor a population geneticist, just to be clear. So I hope you forgive me for any, stupidity I say. My background is in agronomy and botany, and at Kew Gardens I lead a small team of researchers where our focus is trying to understand how plant diversity originated and is maintained through time and space. And then one central question we pursue is why there are some places on Earth, some biomes that are so species rich, where others are so much more depopulated. So one great example is the tropical Andes, as you can see on my screen. And then, as you might have heard from my colleague before, I use orchids as a model organism, especially those distributed in the Asian and American tropics. And so to this end, I combine phylogenetic comparative methods with trait data and characters to understand how different biotic and abiotic variables have driven the evolution of orchids across biomes and timescales. However, more recently, I have been super interested about the almost limitless opportunities that whole genome sequencing data and populations offer to investigate orchid evolution at a microevolutionary timescale. And this is when crops and palms come into stage. So one difficulty of learning population genetics and comparative genomics methods is that when you work with non-model organisms like orchids, then there are not really hundreds of data sets of DNA sequencing data sets sampled in really for population available that one can play with. And so the same can be taught for high quality reference genomes. As Tatiana just explains, there are really few genomes assembled to chromosome level or with high contiguity. But then if you look into crops, that is not the case, right? And so after being really lucky to be involved in a cooperation led by Professor Susanne Renner and Dr. Guillaume Chumiki on the domestication of watermelons, a crop that some colleagues and I thought could be a good candidate to learn more about population genetics and evolution is in very recent timescales, of course, are date palms. And I think that from the oil palm to the coconut palm, dates are perhaps one of the most fascinated cultivated palms. And this is because dates are one of the earliest cultivated fruit tree crops on Earth that played a key role on the subsistence of earlier Asian and African civilizations, because it provides a favorable agricultural environment in an otherwise harsh climate, right? And for this very reason, the historical record of date palms cultivation across time and space is quite rich, and it includes, for example, cone engravings to stone panels. But this also means that then the archaeobotanical record of date palms is really rich and includes, for example, char seeds or items made of date palm leaves, right? And then when you combine these archaeobotanical records or remains with new technologies, like high throughput sequencing, for example, like new technologies, and these enable us to peer into the past of these crops, in this case, dates, and have a more comprehensive idea about its mode and tempo of evolution. So this slide roughly shows us the distribution of date bands and its closely living relatives. The date band belongs to a genus called Phoenix, which is distributed in Africa, Asia, and the Mediterranean. And to the moment, we unfortunately don't have a solid phylogeny for Phoenix, but then many different studies have determined or have found that the closest living relative of dates is Phoenix sylvestris, also known as the sugar date, which is distributed somewhere here. You can see here in blue the distribution of the date band, Phoenix actilifera. And what we understand is that dates were first domesticated into the Arabian Peninsula somewhere like 5,000 years ago. And then there was a subsequent introduction into North Africa. And so when you conduct structural analysis, you can see that there are more or less two discrete populations of Middle Eastern and North African populations of dates. Also, just to say, by the way, that all the research that I'm presenting today has been inspired by the amazing work that Professor Michael Purganam, who just presented some minutes ago, and his team have conducted and with whom we cooperate, right? And one really cool piece of research that was super inspiring for us is this paper published in PNAS in 2019, where they actually discovered that North African date band populations are more genetically diverse. And one reason for such elevated genetic diversity is an apparent processes of gene flow between North African dates and a species known as Phoenix theophrasti, or commonly known as the Crete date band Crete, the date band from Crete, sorry, which is currently distributed in the coastal areas of Turkey and Greece. So in short, it seems that interaction with other species, in particular Phoenix theophrasti and date bands, was important for its evolution. But then one outstanding question was, of course, about the timing of such interactions, right? So keeping in mind this knowledge gap, the goal of our project was to disentangle the genetic contribution of Phoenix species to the date band and the timing of such interactions by means of comparing ancient and modern date band genomes. So how did we do it? We sampled 39 modern accessions of Phoenix actilifera and its closely living relatives, which include 4 species. All this data was produced previously by the lab of Professor Purganam. We also sequenced the genome to some extent of one archaeobotanical remain that I will introduce in the next slides. And then we conducted analysis like genotype likelihoods to derive principal component analysis and interrogation tests, also a mixture. And then we use molecular clocks to try to understand where in time gene flow events took place. So I mentioned that we sampled one archaeobotanical collection for our research, and this is where such specimens come from. Here in Kew Gardens, we have one collection called the Economic Botany Collection, which is open for any research that is interested in conducting research on ethnobotany or population genetics whatsoever. And this collection, which is created by Professor Mark Nesbitt, includes around 400 items, mostly from Egypt and South America, of which 10 are accessions that are made of date band leaves, which are really well preserved. And then this material of date bands has a time series that ranges from 3,500 years to 2,100 years old. And some of this material has been assessed, like the real age of the material has been assessed by means of radiocarbon dating. So we know that it's legit. And the object that we sequence in question is the one that you're seeing here on the top left of my screen, which from now on I will call the Saqqara date band. So this was an item that was precisely found in the animal necropolis of Saqqara. I will introduce this in the next slide. And it's an object that was found to be 2,100 years old, that is made of date band leaves. And the object has a particularly interesting story because at the very beginning, when this object was brought to Kew for research, that was around 1970 or something, the colleague that investigated this object thought that it was an item made for people to rest their heads. So it was called a headpad. And we were actually drafting the manuscript. We went with that hypothesis, and we were asseverating that this object was made at that time for people to rest their heads. But then after discussion with our colleagues from the British Museum, we discovered that there was another object which was is very similar to the one we sequenced, which is the one you see here on the bottom left, right? This object had a better documentation, and actually we discovered that the real purpose of our item has nothing to do whatsoever with people resting their heads, but rather kind of a seal of yards that people would construct at that time to store liquids. So in short, it's a jar stopper that is 2,100 years old. So this is the place where the object was found. Saqqara is an UNESCO site. It's located, I think, 20 kilometers south of El Cairo or something. It's mostly famous for its pyramid of Djoser, which was constructed on the Old Kingdom, which you can see here in this photo. But also has been quite on the news because it has been a site where millions of mummified animals have been found. Right. So in this slide, you can see the outcome of our sequencing experiment of the Saqqara date palm leaf. In short, we produced 30 million reads, of which 30 were dimers, so basically DNA data that you cannot really use for nothing, that has no use. 20% was exogenous DNA, of which we are still trying to understand what kind of DNA is, if it's like fungal or bacterial DNA, if it's ancient fungal or bacterial DNA, which is also interesting. And only about 3% of these 30 million reads was in fact palm DNA. Of this 3%, we found out that 60% was nuclear DNA, about 40%, or let's say 39.5% was plastid DNA, and 0.5% was mitochondrial DNA. So this basically tells us that sequencing archaeological palm remains is challenging. And while this sounds discouraging, the proportion of endogenous DNA recovery, which is this 3%, is comparable with other studies on ancient DNA conducted today. It's not the best, but it happens. And then after seeing this, one big question that one has is how much can be done with this 3% of DNA reads, which translates in roughly 1 million Illumina reads. And the answer might astonish you. I hope it will astonish you. But before I go into that, in order to make sure that the DNA we are using is in fact real ancient DNA and not just modern cross-contaminations, we investigated the damage patterns of this DNA. And we do so because we know that DNA that has hundreds or thousands of years old has a characteristic damage pattern where closer to the end of the fragments, there are artificial mutations that occur. For example, one of the most common ones is C2D misincorporations. So when we conduct the analysis of damage patterns in our ancient sample, here shown in the bottom left, in the bottom of my slides, with damage patterns conducted on modern DNA, which is here at the top of the slide, you can clearly see that the ancient DNA has a high frequency of C2Ds misincorporations towards the end of the fragment, which is something that you cannot really see much on modern DNA. So now we are certain that the DNA we are working with is ancient DNA, right? And precisely because it's ancient DNA, we know that the misincorporations on the data can pose a problem or a challenge for downstream analysis because of these artificial mutations that are incorporated on the data. So after having treated the data and excluded these misincorporations by trimming the ends of the reads, we then investigated the proportion of transitions and misincorporations, transition and transgression rates, which often can help to spot patterns of damage in the data, but also the proportion of error rates in our ancient sample, which is basically an excess of derived alleles when you compare this with an ancient sequence or with an ancestral sequence. And what we found is that the proportion of transition and transversion ratios of our ancient sample, here shown in pink, is comparable to those of modern accessions. And the same can be told for the proportion of error rates, meaning that, in short, misincorporations are not really affecting our downstream analysis whenever the ancient sample or the Sakura date band is included. So now coming back to the question as for how much can you do with 1 million reads? So here is one answer. With that data, we were able to investigate how the Sakura date band is related to modern accessions in a plastid and a nuclear phylogenomic context. So when we placed the Sakura sample in a plastid phylogeny, we found that the Sakura leaf was nested on a clade with high statistical support, was nested on a clade that was made of North African accessions and Phoenix atlantica, which is apparently not a really good species. It might be another North African date band. And this clade in turn was placed as sister to Phoenix sylvestris. And one curious pattern that you see here is that Phoenix dactylifera, or the cultivated date band, which is shown here in blue terminals, is not monophyletic. So it has Phoenix sylvestris nested in. And we think that this could perhaps reveal or tell us about ancient gene flow exchanges between date palms and Phoenix sylvestris. As for the nuclear analysis, what we found is that the date palm is nested in a clade that is all made of accessions of North African and Asian populations of date palms plus Phoenix atlantica. And then this cluster is closely related to Phoenix sylvestris, Phoenix theophrasti, and canariensis. So it's basically online with other studies, previous studies that have shown relationships of date palms with closely living relatives. And now to have a better idea about the ancestry of the nuclear genome of the Sakura date, we conducted a mixture analysis and principal component analysis. And what the admixture analysis tells us, when you assume 4 or 5 ancestral populations, is that the majority of the proportion of the nuclear genome of the Sakura sample is can be traced to North African date palms. So here shown in blue, the bar shown in blue here and here. And then a very small proportion of the genome of the Sakura date palm can be traced to Asian populations, which is shown here in green. At assuming 4 ancestral populations, we were also able to trace introgression with Phoenix theophrasti, and thus supporting the results that Professor Puruganam found in his 2019 PNAS paper. And these results are also in line with the principal component analysis, where our ancient sample here shown in red is placed in a cluster that is close or surrounded by African and Asian accessions, plus Phoenix Atlantic. And then to have an idea whether introgression between the Sakura date palm occur and other closely related species, we conducted this statistic analysis, also known as ABABABA, where we included accessions of Phoenix dactylifera, the cultivated date, the sugar date or Phoenix sylvestris, but also Phoenix theophrasti and Phoenix atlantica. And what we discovered in 3 instances is that the Sakura date palm, which is shown here with this icon, share an excess of derived alleles with Phoenix theophrasti, which is again the Cretan date palm, but also with Phoenix sylvestris, which is the sugar date palm and its sister species, right? Instead of sharing an excess of derived alleles with Phoenix dactylifera and atlantica. This is really exciting because these results are in line with a paper that came just about the same time of ours, also produced by Professor Michael Puruganam, where similar patterns of gene flow between date palm seeds that were germinated from 2,000 years old seeds recover the same gene flow patterns with Phoenix theophrasti, which is really good. So basically what we found out is that gene flow between the Sakura date palm, Phoenix theophrasti and sylvestris already occurred by 2,500 years old. And so to have an idea, a better idea about the timings of these gene flow events, we employ molecular clocks and combination with multi-species quiescent methods. And so the way we did this is that we took representatives of Asian and North African populations of Phoenix actylifera, sylvestris, and theophrasti, and then with these representatives, we sampled all the chromosomes of the genome, and we fragmented each chromosome by windows of 100,000 base pairs. And then we basically feed this data into a relaxed molecular clock and a multi-species quiescent model, which is ideal to infer times of divergence whenever there is gene flow, right? So by doing so across the different chromosomes, we discovered that in every chromosome and genome-wide, there were three main dominant topologies. The ones that you see here, basically. This blue topology reflects the true species tree, or the relationships that we know are like the real relationships in date palms, where Phoenix sylvestris is sister to the cultivated date palm, right? But then, most interestingly, the two other dominant topologies that we found was the gene flow event described by Flowers in 2019 in PNAS, that is basically gene flow between Phoenix theophrasti and North African date palms. But also we found another dominant topology where Phoenix theophrasti was placed as sister to all date palms, all cultivated date palms, which led us to believe or to hypothesize that gene flow might have occurred before 2,500 years old, at least 3 million years ago, right? But then because we know that different topologies across genomes can be produced not only by gene flow, but for example, incomplete lineage sorting, what we did is that to discard the influence of incomplete lineage sorting, we computed the frequency of each possible topology across chromosomes and the genome itself. And what we discovered is that under the scenario of incomplete lineage sorting, you would assume that the frequency of the topologies that you will find will be roughly the same. But in our case, we found that across the genome, there was one main dominant topology, which is the species tree, again, here shown in blue, or this tree here. Then the second most dominant topology was the one here shown in pink, which is the introgression event described by Flowers 2019. And then the third most dominant topology was the one that we discovered here, which basically tells about a more ancient gene flow event between Phoenix theophrasti and the palms that occur about 3 to 5 million years. Meaning that the topologies that we are observing in our genomes are not the product of ILS, but gene flow, right? So to conclude, just about time, to conclude my presentation, the take-home message is that I hope I convinced you today that sequencing archaeobotanical records is still challenging, but once you manage, you can produce really, really cool results and insights. And that we still have to do more advances on how to more efficiently extract and sequence DNA. And for example, approaches like target capture could be more suitable than just basically doing a library and throwing your DNA into a machine, which is what we did. And that our Saquaro date palm is closely related, definitely closely related to Phoenix dactylifera and Phoenix atlantica, which might be the same species. But then these statistics suggest that there was gene flow between Phoenix theophrasti and sylvestris by 2,000 years ago. But then molecular clocks suggest also that gene flow between Phoenix theophrasti and dactylifera might have happened between 3 to 5 million years. I want to thank all my collaborators without whom this project wouldn't have been possible. In particular, discussions with Professor Puruganam, Muriel Baltasar, Jonathan Flowers, and many others were like really helpful. And thank you for your attention.
Thank you so much, Oscar. A very interesting talk. So this talk is open for questions. Do you have questions? Remember to raise your hand in the option of Zoom, please. Any question? Here, Ulises. Now you can speak now.
Yeah, very nice talk, Oscar, and congratulations. It's very nice to see how this project is evolving. I have a question regarding ancient proteomes. Ancient DNA, apparently, according to Enrico Capellini, who has been working on ancient DNA in animals, he says that ancient proteins are better preserved over time. So, and sometimes they are actually very useful to predict some of the very much preserved functions. So I'm not sure, I wanted to ask you if you guys have thought about sequencing proteomes, ancient proteomes as well.
I think that from our side, at least, we are not exploring that possibility yet with our collection. But I know that Muriel Gros-Balthazar, so this colleague here, who is now an independent researcher in Montpellier, I think she is now developing her own program on ancient DNA and deadpan. So I don't know if she's considering that idea. But I would say that this is something like it's definitely worth doing. We don't have the technology here in Kew Gardens to do that, but it would be really cool to do so, especially with fruits and seeds, which we have here stored.
Okay, thanks.
Any other question? In the chat, no? Some other question? Well, it seems like there are no more questions. So thank you so much, Oscar, for your presentation. Very nice.
Thank you for the invitation.
For accepting the invitation. Thanks. So our next speaker today is Susana Magallón. She's coming from the Instituto de Biología de la Universidad Nacional Autónoma de México. And she will tell us about 30 clues to angiospermception and diversification. Thank you, Susana, for being here. It's an honor to have you here. So go ahead.
Well, thank you very much. I am really pleased to have been invited to give a talk. And I think I'm going to use the opportunity to tell you a little bit about some of the research that I have been doing with colleagues and students here at the Institute of Biology of the UNAM, the National University of Mexico. And this research has combined some of the interests along my career, which is studying angiosperm evolution, but also some interesting, I think, techniques on some conventional methods, I think. And my talk, I think, will follow nicely from the talk that Oscar just gave, where he was dating palms from about 100,000 years ago. And I think I'm going to change the temporal scale a little bit. So without further ado, I think I'm going to share my presentation with you. Right.
Yes.
Well, as we all know, flowering plants form an important part of present-day ecosystems. Plants occupied the land about roughly 500 million years ago, and they have acquired a very broad diversity in habit, in life cycles. And throughout most of this history, these plants that have existed are plants that were not angiosperms. So we have a broad variety from liverworts to different types of seed plants. And they involve about a small percentage of extant plant species diversity. However, flowering plants include about some 300,000 living species. That's a very rough estimate. They definitely represent the dominant types of plants, not only in terms of living species richness, but also in terms of morphological variety and function. In particular, flowering plants have evolved a number of attributes that are not found in other plants, some of which we have some clear ideas of where these attributes evolved from. But in other cases, we have no specific ideas of where these organs might have evolved from. Among those, we do not understand very well where is it that the conformation of the stamens came from. And we do not understand the homologies of the carpel, which is the organ that confers angiospermy. And according to some authors, we could consider it as the defining feature of angiosperms. There are also other features, for example, the second integument. But more importantly, in the present day, flowering plants represent the structural and energetic basis of terrestrial ecosystems. This is especially noteworthy in a country like Mexico, where we have a very broad variety of biomes in which, in most of them, angiosperms are structurally dominant. So we can think of angiosperms as an evolutionary Big Bang. We think that angiosperms evolved in an evolutionary frenzy that changed terrestrial ecosystems in a very short time. And as I said before, this has been a wide interest along my career, trying to understand why is it that angiosperms are so abundant and so dominant. And to understand that, we need to take into account the process of biological diversification, which is the underlying cause of biological diversity. And in a very strict sense, diversification is defined as the difference between speciation and extinction. There are many questions surrounding the diversification of angiosperms. For example, whether we had one diversification that characterizes all of the angiosperms versus the possibility that there are several independent diversifications within angiosperms that characterize different groups. Also, we want to know what parts of the phylogeny derived from diversification shifts. We want to know whether diversifications are confined to a particular time period or whether they are spread across angiosperm evolution, and whether all angiosperm lineages are radiating. And finally, if a limit to angiosperm diversification has been reached. To address these questions in this presentation, I will first like to talk about a family-level dating analysis for angiosperms, and then move on to the diversification analyses that we conducted, first focusing on some technical results, and then describing the diversification shifts that we identified. So first, onto dating of the angiosperms. In this figure, I am summarizing some of the estimates of the age of the crown group of the angiosperms obtained by molecular clocks from the earliest attempts to more recent attempts. And as you can see, the estimates of the age are very broad. Some of them, in particular, for example, have very wide confidence intervals associated to them. And these estimates derived from molecular clocks, which are here in red, contrast very widely with estimates derived from the fossil record, which are here. I am showing the stratigraphical period of appearance of different angiosperm orders during the Cretaceous. And as you can see, there is basically no overlap between the molecular estimated ages and the fossil record. So this is one classic example of a rock versus clock conflict. To estimate the timing associated to angiosperm diversification, we conducted molecular clock analyses in which the taxonomic sample included about 800 species that represent about 87% of living angiosperm families. But even though the representation of angiosperm families is not 100%, these families include about 99% of living angiosperm species, and all of the orders are included. We used a conventional molecular data set, We estimated phylogenetic relationships with maximum likelihood. And this study is very interesting because it included two different approaches to calibrating the tree. This part of the study is usually underappreciated, but I think it's very interesting because the first approach to calibration that we used is using a method that is derived from quantitative paleobiology, in which we estimate a confidence interval around the age of one of the nodes in the phylogeny. The node in the phylogeny for which we estimated a confidence interval is the crown node of the angiosperms. And to estimate this method, we used a method proposed by Charles Marshall some years ago, which, as I said before, is derived from methods that are used in stratigraphy to estimate the real time of origin and of extinction of a lineage in the fossil record. The youngest age of the confidence interval is given simply by the age of the oldest fossil that can be reliably assigned to that group. And the maximum age of the interval, or the oldest age of the interval, is given by this equation in which some critical information is n, which is the number of branches in a phylogenetic tree which are represented in the fossil record. In our study, we used a tree at the family level from which of the living angiosperm families, 123 are represented in the fossil record. So the fact that this number is very large had a very important influence in the size of the confidence interval that was estimated. The other important factor is age, which represents for each branch represented in the fossil record, in how many fossil localities it is found. And that information is very difficult to obtain. So therefore, we simply used 1, which is a huge underestimate, and that will allow for ages to become older. We also used a 95 confidence interval, and by applying this equation to the data in our tree, we obtained a confidence interval for the crown node of the angiosperms between 136 and 139.35 million years. In addition to this method for bracketing the crown node of the angiosperms, we conducted a conventional molecular clock in which we obtained calibrations from the fossil record to provide minimum ages to internal nodes in the phylogeny. We obtained this information from a data set that was compiled in my lab that includes more than 3,500 phylogenetically organized angiosperm fossils, and from which we critically examined to identify which fossils could calibrate internal nodes in the tree. From this, this is the phylogram. Here you can see all the terminals, and you can see all the internal calibration nodes that were directly derived from the fossil record as minimum ages. And we combined that with a calibration on the crown node of the angiosperm that provided, this was introduced as a uniform distribution between the bracket that we obtained with the paleobiology quantitative method. This is, we estimated the dates with BEAST, which uses Bayesian inference to simultaneously estimate phylogenetic relationships, divergence times, and molecular substitution rates. And the timing of speciation is derived from a birth-death process that generates the tree, and also from the node calibrations that were implemented as prior parametric distributions. This is the dated phylogenetic tree from which we can obtain times of origin for almost all angiosperm families, which has been used now in a number of different analyses to investigate angiosperm evolution. And as an example of that, we, together with colleagues from different universities, we used that tree to estimate the structural attributes of the flower of the most recent common ancestor of all of the angiosperms but also, as in this example, to do an ancestral state reconstruction of a number of attributes. And in this example, this is the reconstruction of functional sex. But let's recall that we are interested in angiosperm diversification, and we are interested in addressing these questions. So to investigate the rate diversification of angiosperms, we conducted BAM analyses, which is the acronym for Bayesian Analysis of Macroevolutionary Mixtures, which was proposed by Dan Robowski in 2014. Basically, this method uses a compound Poisson process that identifies shifts in diversification rate among phylogenetic branches, which are here shown as these dots, and also changes on the rate of diversification along each phylogenetic branch, which is what you perceive as different colors along particular branches. BAM does this by generating a posterior distribution of so-called shift configurations, each of which represents a combination of the number of shifts across the tree and their phylogenetic and temporal placement. This method became very popular a few years after it became proposed, and many researchers were using it to investigate diversification in different clades. But a few years later, Brian Moore and collaborators published a study in which they expressed some concerns about the performance and the accuracy that could be obtained with BAM. This criticism was responded by Robowski and collaborators, and I am going to summarize one of the main concerns expressed by Moore et al., which is the observation that the compound Poisson process used by BAM is basically unidentifiable, and therefore the posterior estimates of the number and the placement of the shifts in diversification that BAM produces very much mimic the prior distribution. And Moore et al. Produced these distributions where basically the prior and the posterior are completely indistinguishable. Robowski and collaborators re-examined Moore et al.'s data, and they basically obtained very different results. Here you can see the results obtained by Robowski, where you can see the prior, which is in red, which is distinctly different from the posterior, which is in gray. So Robowski summarized the results, saying that the prior and posterior distributions that can be produced by BAM are distinctly different, but very importantly, that users should consider only those shifts that are very highly supported by the data. In other words, by the posterior distributions. So we took into consideration those recommendations, and to conduct our diversification analyses, we used the data tree that I have already presented to you, and we conducted not one, but actually 6 different BAM diversification analyses using different priors for the number of shifts across the tree. The general recommendation is that if your tree is small, you should use a small number of shifts. But if your tree becomes larger, then you should use as prior distribution a larger number. These are in logarithmic values. So I'm going to show you the results that we obtained from the prior and the posterior distributions. Here you can see the prior distributions are in blue, the posteriors are in orange in our 6 analyses, and you can clearly see that the prior and the posterior are different in all cases. We conducted all of the analyses under the 6 different prior distributions, and then we ended up with a problem because we actually had 6 different analyses, and it was not very clear which one to choose. So the solution that we chose is that from each analysis, we identified the shifts that were most strongly supported by the data. And then we compared the results among the 6 analyses. And by doing this, we identified a set of strongly supported shifts in each analysis that were at the same time shared among the 6 analyses. And this gave us a set of strongly supported shifts that were shared among all of the analyses which allowed us to summarize the 30 core diversification shifts across the angiosperms. I would like to mention, just technically, that in some cases, the shifts are found as a single shift in one branch that is highly supported, but in other cases, the shifts can be found in adjacent branches along the tree, each of which has a low support, but together they have a very high support in this phylogenetic region. So we found all those two kinds of instances. This is the main result. This is what the BAM results look like. They provide this graph, which is called a phylorate plot, in which, well, it's basically a phylogenetic tree in which each of the branches has a different color, and the color can change along the branch, indicating the estimated rate of diversification, which follows this heat map, with warmer colors indicating higher rates. And a phylorate plot can also be obtained for the rate of speciation and of extinction. So this is the phylorate plot for all of the angiosperms. It's possible also to estimate the rate of diversification in blue, speciation in orange, and extinction in green through time. I'm going to show you now the shifts in diversification. This is for the angiosperms as a whole. Before going to the shifts, I would like to mention that the indicated rate of diversification does not show that angiosperm diversification is declining towards the present. Actually, it's increasing a little bit. So looking at the shifts, the first shift corresponds to mesangiospermy, which is a major clade within the angiosperms that includes all of the angiosperms except the anagrade. And this, the shifts, I will be presenting them to you in chronological order. So this is the oldest shift. The second oldest shift corresponds to a clade including Vitalis plus Rosids within the eudicots, actually within the pentapetalae, followed by Fabidae, also within the Rosids, then Ericales within the Asterids, Myrtales within Rosids, a clade within the monocots that includes Aracales, Gingiberales, and Camelinales, then Ranunculaceae, family Ranunculaceae within the eudicots but outside pentapetalae, Lamidae, which is a large clade within the Asterids, family Fabaceae within the Rosids, another large clade within monocots that represents the sister group to orchids, then Sapindales within Rosids, Polygonaceae plus Plumbaginaceae within the Caryophyllates within eudicots, approximately family Asteraceae within, of course, the Asterids, the Dipsacales also within Asterids, a clade formed by families Montiniaceae, Hydroliaceae, Sphenocliaceae. Probably you've never heard of these families. Actually, this represents the only shift that we detected that represents a decrease in diversification. These three families are very small. Family Orchidaceae, Crassulaceae within eudicots, within the Superrosids, Moraceae and Urticaceae also within the Rosids, approximately Apiales within the Asterids, Cyperaceae and Juncaceae within monocots, Euphorbiaceae within the Rosids, Campanulaceae within the Asterids, Celastraceae also within Rosids, actually within Fabids, Amaranthaceae within Caryophyllales, within the eudicots, Piperaceae, the only detected major shift within Magnoliids, another clade including Brassicaceae, Cleomaceae, and Caparaceae, also within the Rosids, within Malvids, Poaceae within the monocots, Cucurbitaceae also within the Rosids in Fabids, Malvaceae within Malvids in Rosids, And finally, the last one, Cactaceae within Caryophyllales. So these are the 30 major diversification shifts that we detected within the angiosperms. We find, you can see that these diversifications and depletions are overlapped through angiosperm evolution. They are not characterized in one particular group. We find, although this needs to be explored in greater detail, possible different diversification dynamics in rosids and asterids. In one of them, in which there is one big radiation at the onset of the clade, and whereas in the other one, there seem to be several independent radiations within this large clade. And it's possible to identify a number of lineages that are species poor, which represent either ancestral slow rates of diversification versus decreases in the rate of diversification. To summarize the results, we estimated the timing of angiosperm evolution in a study that has served as a landmark for independent and more recent timing relaxed molecular clock studies. For example, this is a result that we produced with colleagues from UNAM, but also from other universities in different parts of the world. And also they have served to conduct different evolutionary studies. For example, a study in which we investigated and estimated the structural attributes of ancestral flowers. In terms of diversification analyses, we provided what I think are interesting technical results, indicating that different prior values for expected number of shifts resulted in distinct prior and posterior distributions, but basically, importantly, a highly overlapped set of core diversification shifts. Regarding our investigation of angiosperm diversification, we find that the extant species richness of angiosperms can be explained by a number of independent diversification increases, that they occurred in different parts of the phylogeny independently, that they took place in a period of over 100 million years throughout angiosperm history, and therefore are not concentrated in one particular point in time. While several different lineages of angiosperms are diversificating, others are decreasing in the rate of diversification. And it seems that a limit to angiosperm diversification has not been reached, but it seems that different clades are taking different turns, if we may put it that way, into diversifying in different points in time. I would just like to finish by mentioning a few questions that we obtained from this study and possible future directions. And we confirm the idea that the causes underlying high diversification are most probably linked with the relationship among complex morphological and phenotypic attributes, different relationships among lineages, and also different physical conditions in different points in time. Also, it's possible that different major clades within angiosperms are characterized by different diversification dynamics, something that needs to be explored in greater detail. And finally, that another question that seems to me very interesting is try to understand why is it that some lineages are very ancient and very species poor, because according to different models of how these clades can evolve, it's basically very difficult to exist for a very long time, having a very low rate of species diversification. So with that, I would like to conclude and take questions if there is time. But before that, I would like to thank my students and collaborators for what has been a very long and productive time of research with them. Thank you very much.
Thank you so much, Susana, for so beautiful talk. I learned a lot. So let's see, we have questions. Mr. Ulises, you can speak now.
Yes, thank you, Alejandra. And thank you, Susana, for very, very nice talk. It caught my attention that, well, you were talking about the diversification shifts. And for many of us here in the audience, I mean, we are lovers of Arabidopsis within the Brassicaceae, potato and tomato within the Solanaceae, and Fabaceae for legumes, right? So I was surprised that, you know, within your results, you didn't find any, I mean, hopefully I didn't miss these families, but you didn't mention the diversification shifts. On the other hand, you did mention Cactaceae, which I'm very happy because I love cacti and working on them. But regarding these model species in Brassicaceae, Solanaceae, and Fabaceae, what can you tell us about the possible implications for evo-devo studies that we might conduct? I mean, is that something that are we working on the wrong species, or should we just move to another set of species to study evo-devo, or what is your take on that, Susana?
Well, I think that the lineages that you mentioned, they were included. Please recall that this study is at a very wide phylogenetic level. So I did mention that there was an increase in diversification associated to Brassicaceae, Caparaceae, and Cleomaceae. So that one, I can check that one. Also for Fabaceae, it was one of the earliest. We had one for Fabidae, which is a very large clade that includes the legumes, the nitrogen fixing clade, and many others. And so, but also I mentioned Fabaceae, which the thing in Fabaceae is that the shift is not clearly associated to the onset of Fabaceae, but it could be one node before or one node within Fabaceae. So that's an interesting one. I don't remember which is the other one you mentioned. Was it orchids? And
Brassicaceae, Solanaceae, and Fabaceae. I think they are the ones I mentioned.
Yeah, okay, Solanaceae. I don't remember exactly Solanaceae, but this is the thing that it seems that there is a shift that characterizes all the Solanales and the clade that include the Solanales. So it may be that the Solanaceae, in fact, by themselves, do not represent a shift in diversification, but actually the shift in diversification came before in a clade within which the Solanaceae is nested. So that's something that needs to be looked at. And this is what I was mentioning, that one question is whether in different large clades, diversification rates characterize very large clades as a whole, or the diversification rates are associated to particular clades within a group. And it seems that if you compare rosids and asterids, they have very different approaches to doing that. And I think that is very interesting because that would be a very interesting macroevolutionary result that you could only obtain by looking at angiosperms as a whole at this level.
If you allow me a second question, I wonder what is the relationship with genome size evolution with these diversification shifts?
Well, that of course is a very interesting question. I have not worked on that directly, but I know that there are a number of papers that have been published to address that. For example, there is a paper that looks at not diversification, but actually species richness. And there is some kind of a link between genome duplication and very high species richness. And in fact, this difference does not occur on the same phylogenetic branch, but there is a time lag because you have the genomic duplication, and a few nodes further in the tree, you have the increased numbers of species. And the authors have argued that actually this is consistent what you would expect from having a gene duplication where you have a whole set of genes with which you can, as a plants can do a lot of experimentation, acquire new functions, and then the species richness would follow. So that's there. Some authors have actually found a link between genome duplication and high species richness.
Okay, so the next question would be for Aida Martinez. So you can speak now, Aida.
Yes, do you hear me?
Yes.
Thank you. Thank you, Dr. Magallon. I know that this is not the focus of your study, but what is your opinion about crops? Is there evidence that the modern agricultural practices are really diminishing the biodiversity of wild relatives? Or deforestation is most important? I don't know.
I think that the only way that crops can diminish natural variety is by finishing with natural environments, but
Yeah.
not per se. But I think that a crop per se does not diminish natural variation. I hope I am making my opinion clear about that. So it's not the crop per se, but the fact that by cultivating widely, humans are finishing with natural environments.
Yes, but not the crops or the
Not the
fertilizers.
that's not my field. So
Yes, I know. Just your opinion. Thank you very
that
much.
would be my unqualified opinion.
Okay.
Thank you. So Alma Piñero, go ahead.
Yes, hi, Susana. Thanks for your talk. I was wondering, I know that your study it's the macroevolution of angiosperms, but I was wondering, what do you think would be the lowest resolution? Where's the limit in where you can define diversification events that have maybe occurred after the biggest shift you documented? Like, what would be the resolution? Like, how closer to the present can we go and infer either diversification or real declines in diversification in different plant lineages?
Well, one way to do it is perhaps using the type of studies like the one I presented to identify groups and then separate that group and conduct studies on that group. And the way I would do it is by increasing very much the sampling of living species. I think that is the key. I think the way to do it would be by increasing the sampling of living species. But then you face other limits, which have to do with the computers being able to deal with vast amounts of data. And especially if you do Bayesian analyses, sometimes the chains do not converge. So I think the limits are both in terms of the, not so the method, in terms of the model, the mathematics seem to be very solid, but the ability for the statistical methods to converge, basically Bayesian methods. You can also do maximum likelihood estimations, but they also have some drawbacks. And the other thing that is very important and might be a limiting factor is the ability to get a sample of all the living species in one particular clade, especially if you want to do it at a broader level. Maybe if you're looking at a genus, it might be doable. But then if you're looking at a family, and this is a large family, for example, Fabaceae or Brassicaceae, it might become more challenging. But I think that computer power is increasing. And I'm very hopeful that in the future, we will be able to do these analyses at a more detailed level.
Okay, thanks. So let's take the last question because we are going into a break period. So Oscar, you can speak now. Oscar Perez.
Yeah, thanks very much, Professor Magallon, for such insightful talk. I had a couple of tricky questions. Which is your view on Daniele Silvestro's 2021 paper on the pre-Cretaceous origin of flowering plants, and how important is the definition of angiosperms versus flowering plants on understanding, their timings of diversification? Thanks.
Well, man, I have about half a minute to respond. I have a opinion on that. Basically, where do I start? I'm going to start with the data. They took the data from the paleobiology data set. So it's problematic because you start with the assumption that every name there corresponds to a species, which is, well, that's not the case. Second, just looking at the results, you can see that sister species or sister clades have widely different ages. So how can you explain that? You would have, because their method is phylogenetic free, but if you look at the phylogeny, you will see that sister clades have widely different ages. So that's something that I really cannot explain. How do you define, well, how do you define angiosperms? Well, there's a very long branch separating angiosperms from their extant living group, which are the acrogymnosperms. And along that branch, you had a number of apomorphies evolving. So which one do you want to choose to define the angiosperms? I think all of them, but then that makes things complicated. And if you really make me choose one, I would choose the appearance of the carpel. But that's my opinion, and I know many colleagues have a different opinion. Yeah. So it's problematic.
Thank you very much.
Thank you, Susana, so much for your presentation. It was really beautiful. And
thank you for inviting me.
Thanks. See you soon. So let's go to the break period. So please come back at 11:45 for the next talk of Natalia Pavón Mora. See you soon. See you.
Our next speaker is Natalia Pavón Mora from University of Antioquia, Colombia. She's going to talk about genetic mechanisms controlling flower and fruit diversity in non-model neotropical angiosperms. Go ahead, Natalia, and thanks for accepting our invitation.
Thank you, Felipe. Thank you, everyone. Hold on one second. Let me just start my video and I will share my screen.
Yeah.
Okay, I think it's good now, right?
Yep.
Okay, thank you so much for this invitation. I'm very excited to be here with you today, sharing a little bit of the research that we do on the genetic mechanisms controlling flower and fruit diversity in non-tropical angiosperms, in neotropical angiosperms. So hold on one second, let me get my pointer. And so I just want to start by explaining a couple of ideas based on key factors that basically our research that is framed upon these two key ideas. The first one has to do with the fact that most angiosperms are concentrated within the chorioidicots. And this is interesting because that basically suggests that 75% of all angiosperm species are concentrated into this single group. However, one can claim that most of the floral diversity is actually concentrated outside of the chorioidicots. So at least in terms of the floral ground plan, we can see a lot of variation in the basal eudicots, the monocots, and the basal angiosperms. Now, a second idea that I want to highlight is the high occurrence of whole genome duplication events during flowering plant evolution. And so here, basically, I want to point out to you that most of those whole genome duplication events are concentrated again here in the chorioidicots and in the monocots. And basically this renders our knowledge from model organisms, say like Arabidopsis, tomato, or grasses, very difficult to extrapolate to other flowering plant groups. And this is true for both the genetic complement, but also it's true for a gene function. And this is especially important for plant lineages that lack most of those whole genome duplication events. So during the past 9 years in the lab that we run in the Universidad de Antioquia, we have tackled a number of developmental biology questions using our biodiversity. And so we have tackled, for instance, genes involved in flower and fruit development in the chloranthacea. Do recall that this plant lineage lacks most of those whole genome duplication events that I just described. We have also been studying genes involved in floral organ identity, symmetry, and color evolution in the Aristolochiaceae. And again, in a similar evolutionary scenario, lacking most of those whole genome duplication events. More recently, we have started to investigate genes involved in flowering time in native orchids. And this is a completely different scenario because not only Orchidaceae have the monocot duplication events, but also family local duplication events. We have tackled the genetic basis of Apetalii in Boconia frutescens that, unlike its relatives, it's a tropical poppy. We are currently studying genome content in a miniature parasitic plant, which is Pilostylis in the Apoanthaceae in the order Cucurbitalis. That's already in the chorioidicots. In the chorioidicots as well, we have been tackling genes controlling floral symmetry in the nasturtiums within the genus Tropiolum in the Tropiolaceae in the same order as Arabidopsis. And we are also interested in studying the genetic basis of fruit diversity in both the Solanaceae and the Rubiaceae. So because of the time limitations, I will only share with you today 3 of these projects, and I will start right here in the order Piperalis. Here we have concentrated our attention in the Aristolochiaceae family, which comprises all of those species in the order with perianth. So as you can see right here, perianth is extremely variable in this family. It's biseriate in Saruma with 3 green sepals and 3 yellow petals, and it's uniseriate and sepal derived in all other genera in the family. However, the perianth diversification or diversity reaches its peak in the family in the genus Aristolochia. As you can see right here in this slide, it varies in shape, size, colors, symmetry, among others. So early floral developmental stages in Aristolochia species confirm that the perianth corresponds to 3 sepals. Never have we seen petal initiation, and despite the fact that there are no petals, we believe that there is a transfer of function from petals to sepals, as sepals often display colors others than green in the ranges of yellows and purple. But it also has elaborations in the epidermis, usually related with the formation of floral trichomes that help to attract but also trap the pollinating flies. Now, all species in Aristolochia share an additional feature, which is the congenital fusion between the stamens and the stigmatic tips in a highly unusual radially symmetrical gynostemium. So here we have asked a number of questions regarding floral organ identity in Aristolochia, the genetic mechanisms underlying floral symmetry in Aristolochia, but also the genetic mechanisms controlling pollination-related specializations, and particularly trichome formation. So in this case, we usually start with reference transcriptomes. And in this case, we started out by assembling a reference transcriptome for Aristolochia fimbriata, which is a small-sized species with very fast life cycles, easy to grow in the lab. But at this point, we have gathered at least 11 reference transcriptomes from different species. In the family, we usually search the homologs for a particular developmental process that we're interested in studying. We usually follow up with additional phylogenetic analysis to confirm homology of these genes, and we follow up with detailed expression analysis using RT-PCR, qRT-PCR, in situ hybridization, and more recently, large-scale comparative expression analysis. So I want to give you an example. We started out by tackling the genetics underlying floral organ identity in Aristolochia. And for that, we took as a reference the model, the ABCDE model of floral organ identity established in Rhabdopsis, which describes basically how 4 different classes of transcription factors come together in an overlapping manner to promote the identity of each of the floral organs. And so for that, we started out by isolating all the homologs for the model from the reference transcriptome of our target species, Aristolochia fimbriata. And then we followed up with a very detailed expression analysis. So we did the analysis for the C and the D class genes, the B class genes, the A class genes, and I won't tell you a lot about this data, but I do want to highlight our most important result from this research. And that was the fact that we did find an AGL6 homolog that is specific to perianth identity that is maintained during perianth differentiation in this taxa. And importantly, this AGL6 homolog interacts with Sepalata2, which we tested by protein-protein interactions using yeast-2 hybrid. So basically, this allowed us to postulate AGL6 as a key player in sepal identity, in perianth identity in these magnolids, in a fairly modified model when you compare it to other monocots and basal eudicots and core eudicots. More recently, we started to investigate the genetic basis of floral symmetry in Aristolochia. Here, the model has been framed in Snapdragon. In Snapdragon, what happens is that Cyclodia and Dichotoma, which are TZP BHLH transcription factors, are turned on in the dorsal side of the flower. And basically, they activate radialis, which is a MIP transcription factor. And radialis outcompetes the varicata, displacing this transcription factor to the ventral portion of the flower. Now, when you look at expression patterns, Cyclodia, Dichotoma, and radialis are usually concentrated or always concentrated to the dorsal portion of the flower, lacking in the ventral region of the flower. And whenever Cyclodia and Dichotoma are mutated, the flowers not only are radialized, but also become ventralized. So the question here was whether cycloidea genes were also controlling bilateral symmetry in Aristolochia. Do recall that bilateral symmetry is an unusual feature in this group, in the magnolids. In fact, in basal angiosperms, it doesn't occur. It only occurs in Aristolochia. So here the question has to be slightly rephrased. Aristolochia does not have true cycloidea 2 homologs. It only has cycloidea genes, which predate all of the choriodicot specific clades, which include cycloidea. So we started out by mapping expression of these cycloidea-like genes in 2 different species, Aristolochia arborea and Aristolochia fimbriata. And unlike what is expected from a factor that is controlling a bilateral symmetry, what we saw was a homogeneous expression of these genes throughout flower development from the early stages to a late perianth differentiation, suggesting that these genes were not important to control bilateral symmetry. And this was again confirmed when these genes were overexpressed in Arabidopsis, and they don't show any particular features regarding changes in petal size, which is an estimation of the role in bilateral symmetry. So basically, we wondered whether other factors were important for establishing bilateral symmetry, and we turned to the Cincinnatta genes, not only because they are the closest relatives, phylogenetically speaking, but also because from an independent parallel study aiming to identify differentially expressed genes in different floral portions of the flower of Aristolochia ringens, we realized we continuously kept picking up Cincinnatta genes as upregulated in the limb, which is the distalmost portion of the flower. And in fact, when we followed up with a specific in situ hybridization studies, we did see, as you can see over here and over here, that these genes were concentrating predominantly in the ventral portion of the flower, and particularly in the limb. This is true for 2 out of 3 homologs of Cincinnatta. Now, importantly, when we did the studies of histone 4, histone 4 is a marker for cell division. We discovered that the expression of Cincinnatta genes overlaps with the expression of histone 4, which suggests then that Cincinnatta positively regulates cell division, which is a process that had been already been reported in the flowers of snapdragon. So perhaps in a cycloidea-centric universe, we have been missing some of the contributions of other TCP genes outside of cycloidea, like including Cincinnatta and PCF factors. And we know this is true at least for Orchidaceae and for the nasturtiums. We have picked up a number of these genes that are differentially expressed between the dorsal and the ventral regions of the flower, suggesting that perhaps these genes could be more strongly explored across angiosperms. So regarding the genetic mechanisms controlling pollination specializations, I just want to highlight a couple of lessons that we have learned from our most recent research on trichome development. And the first thing that I want to highlight is the fact that the highly diversified trichomes inside of the flower of Aristolochia only use a small subset of the key regulators when compared to those genes that are active in order to form trichomes in the leaves. So that's, to us, was really interesting. The other thing that I want to highlight is that by doing large-scale expression analysis using 2 different species, one with trichomes and one without trichomes, we were able to discover that positive regulators in Aristolochia for trichome development resemble more closely those of cucumis than those characterized in Arabidopsis, which means that then the genetic regulatory network for multicellular trichomes shares some common hubs in distantly related angiosperms. Okay, so let me now move on to our research in holoparasitic plants. Here we have concentrated our attention on Pilostyles boyacensis, which you can see right here in the bottom of the slide. Pilostyles boyacensis is 1 out of 11 species of the genus Pilostyles, which grow in dry habitats around the world. Pilostyles is also an endoholoparasite that lives inside of the stem of Dallea quatrecasaceae, which is a member of the Fabaceae family. And this is very special relationship is found in high Andean thickets. So being an endoholoparasite means that your entire life cycle, or most of it, occurs completely inside of the host. So you can see that by these photographs, and here in this anatomical section and in this micrograph, you can see how all of the cells of the endophyte grow inside of the host, and then eventually even floral development occurs inside of the host. And you can only see the plant, it becomes evident when the fully formed flowers emerge through the host stem. The vegetative growth is very difficult to detect. There are no true root or shoot apical meristems in this plant. However, the flowers are very obvious, as you can see in these pictures. And here in the left, you have the male flowers. Here in the right, you have the female flowers. And right after ovule fertilization, those female flowers form fleshy fruits, berries, carrying a ton of minute seeds with fully formed embryos. So such a unique system calls for a large number of questions, but we have so far focused on 3 of them: genome evolution and gene content in Pilostylis boyacensis. We want to understand what should apical meristem related genes are retained under such extreme vegetative reduction in this plant. And we are also interested in understanding the developmental gene network for flowering and floral organ identity. So for this, we have generated genomic data using 2 different strategies, but we have also generated reference transcriptomes for the entire life cycles, or spanning the full life cycle of Pilostylis boyacensis. And so for that, we have generated one that is a mixed tissue between the endophyte and the host. A second one specifically from flowers that have emerged through the host stem but prior to anthesis. And the third one specifically from fruits with the seeds inside. So at this point, we're starting to sort out the content of this metagenome. It has not been an easy task, but we already sequenced and published the plastome of Pilostylis boyacensis. As you can see, it's highly reduced. It's only 12,000 base pairs. We are in the process of assembling the mitochondrial genome, and we have a rough genome estimated to be around 300 megabases. So perhaps more important, at least to me, or more interesting, are the developmental questions. And so here you have an example of a plant that, again, does not have root or shoot apical meristems. And we started to characterize which of the genes that were proposed as canonical for pluripotency in Arabidopsis were still maintained in this unique plant. And so in Arabidopsis, WuSCHL is a key transcription factor that controls pluripotency. It maintains stem cell identity, and then WuSCHL activates its own counteracting factor, clavata3 and clavata1. And this fine-tuned feedback loop is actually reinforced by the presence of BAM. BAM stands for barely any meristems, and WOX9, which is a WuSCHL homolog. So again, the question is, from this canonical network, how much of it is conserved in Pilostylis? And so what we found was actually quite remarkable. Pilostylis boyacensis has a WuSCHL, but it doesn't have it in earliest stages. It only has it expressed in intermediate stages of flower development. It also has a WOX9 that's actually broadly expressed during the life cycle. It does not have any clavata3 or clavata1 homologs, but it does have BAM homologs, and its presence is quite rare because given that their role is to quench out clavata3 excess, then it's unclear what it's doing in the absence of clavata3. So basically, this has led us to conclude that there is a substantial reduction in the transcripts linked to shoot apical meristem in this plant. We do know that Pilostylis shows at least one transcript with all functional domains for BAM1, WuSCHL, and WOX9. There are no clavata3 or clavata1 homologs that we could identify. And that means that whether perhaps there are alternative repression modules for the pluripotency genes, or there is no need for repressing them as there are no lateral organs being formed. And then our third conclusion is that the minimal genetic regulatory network, we think, at least trying to extrapolate it from the canonical data established in Arabidopsis, the minimal genes that this holoparasite requires would be WOX9 and BAM1, given that these are the only genes present in the earliest stages of the endophyte. So we wanted to test if that was true or if similar trends were found in the gene network for flowering, floral organ identity, and fruit development. And so what we did was actually trace all of the flowering integrators, again, using all of them, including those positive regulators like FTA, GL24, SOC1, and LEAFY, but also trying to identify the homologs for the negative regulators. And we did the same task for the floral organ identity genes that I already showed you before. So here, the results are very different. The evolutionary reconstructions of all of these lineages, including the PVP genes and the Matchbox genes, show extensive usage of host genes, meaning that every time we found a transcript, most of those transcripts were actually nesting within Fabaceae homologs. And so basically this tells us something that is very different from what I showed you before. Now, conversely, floral meristem identity genes like LEAFY and floral organ identity genes, like all of the Matchbox genes that I just showed you before, are primarily endogenous genes, meaning that they don't cluster with Fabaceae. So basically, this has led us to suggest that all flowering promoters so far identified, including FTA, GL24, and SOC1, likely belong to the host, but are being actively used by the endoholoparasite. And this is not unusual, at least this has been shown to occur for Cuscuta and specifically for the FT genes, which are small mobile signals. The same is true for SBP, the only flowering repressor identified, but different from that, all the floral meristem identity genes and floral organ identity genes are endogenous in Pilostyles. Okay, so I think I have a couple of minutes still, and I want to show you a little bit of the research that we have done on the genetic basis of fruit diversity in the Solanaceae and in the Rubiaceae. So fruits, strictly speaking, are extreme ontogenetic changes of the carpel wall after ovule fertilization. You can see how they vary morphologically speaking during that unique transition, and this is also accompanied by anatomical changes, sometimes extreme anatomical changes. And because fruits are the ontogenetic result from carpels or from the carpel wall, they are, of course, strictly found in angiosperms. They have evolved around 150 million years ago, but you can see how they display one of the most outstanding variation in form, in shape, and in seed dispersal strategies, as you can see from these photographs. Now, the genetic basis for fruit patterning and dehiscence have been identified in Arabidopsis, and in the system, or in Arabidopsis, is known that fruitful regulates the cell division in the fruit wall. Replumulase is important for the formation of the replum right here. Between the two of them, they regulate or negatively regulate shatterproof, and shatterproof is key for promoting the identity of the dehiscent zone. Now, shatterproof activates in the dehiscent in the lignified layer, and it activates in the dehiscent together with alcatraz and spatula in the parenchymatic and lignified layer. And on top of that genetic cascade is APERLA2 regulating the entire network. Now, at this point, it shouldn't be a surprise that all of these genes belong to different gene families. And despite the fact that we know a lot about this genetic network in Arabidopsis, we still don't know a lot about how it behaves in different non-model species. And so our lab has concentrated the attention in three major questions. The first one is the evolution and the changes that this genetic regulatory network may have had in different angiosperm groups. The second one is whether there are genes that can fine-tune anatomical changes during pericarp maturation in non-model species or outside of Arabidopsis. And we're also curious about the plesiomorphic roles of those genes that are not angiosperm exclusive. And so I just want to highlight an example of what we have been doing in the Solanaceae and the Rubiaceae. We have been, for the past 5 or 6 years, collecting fruits that represent the diversity of fruit types in those two families. And at the same time, we have generated reference transcriptomes. Every time we collect something, we generate a reference transcriptome for it. Now, do keep in mind that the Solanaceae has superior ovaries, whereas inferior ovaries are the rule in the Rubiaceae. So we have followed up with comprehensive, very detailed anatomical analysis, and I just want to kind of briefly summarize our results just by telling you that every time we see a superior ovary-derived pericarp, changes in anatomy are fairly easy to identify. So basically, we can say that lignification only occurs in fruits that will dry out and dehisce, and most of the time, at least in the endocarp, and most of the time, cell proliferation and cell expansion are limited or primarily occurring in fleshy fruited species. This is not the case for the Rubiaceae, as they are inferior ovary derived. So basically, we see dramatic changes in fruit anatomy that are very hard to pinpoint in terms of their origin. Sometimes those changes come from carpellary tissue, but sometimes they come from extracarpellary tissue. Now, something very interesting that we have observed is that the relatively simple fruits of Solanaceae carry a dramatic genetic load due to Solanaceae-specific duplication events in all of the gene lineages that are important for fruit development, whereas the relatively more complex Rubiaceae fruits have simpler or use simpler genetic complements. This is the same that I just showed you, only I'm showing you here a view of their expression. And here you can see how, again, simpler fruits require numerous active genes, whereas more complex fruits require less genes active. And something that I want to highlight is that we still don't know a lot about the transcription factors that contribute to those changes in extracarpellary tissues. I just want to finish up by telling you that we have standardized tools to test gene function. We have used virus-induced gene silencing, at least here I'm showing you those data from the Solanaceae. We have done this for the positive controls that you can see in this slide, but we have also targeted specific genes that we believe are responsible for those subtle changes in pericarp. So for example, we have studied spatula and alcatraz genes, and we know that these genes are important for repressing lignification, both in the dry dehiscent fruits as well as in the fleshy fruits. And so at this point, we're in the process of trying to take that technology and transfer it to Rubiaceae members. So with that, I want to thank all our funding sources, both international and national, our national collaborators, our international collaborators, which have facilitated and helped in any way they can in this research. And here I just want to show you the amazing team that we have. These are the people that we have been able to train in a primarily teaching institution. And I hope there is time for questions, but if not, feel free to reach out by email or peruse our website. Thank you.
Thank you very much, Natalia, for your very, very interesting and colorful talk. More questions? So, is there some? There is a question. Two questions. Yeah, go ahead.
Oh, that's really amazing work. Thank you very much, Natalia. My question is not directly related, maybe, but you mentioned that Aristolochia has a short life cycle. What is the reason for that?
Not actually all species of Aristolochia do that. Aristolochia fimbriata it's a small-sized kind of smaller compared to all other species. There are some that reach, well, so most of these are lianas, so they actually reach bigger sizes than what I just showed you for the Aristolochia fimbriata. This particular species is small, it's herbaceous, and a fast life cycle in this case would be from seed germination to seed set around 3 months under lab conditions.
Oh,
and that's
okay.
why it was selected. So it's not super short, but okay. It's definitely shorter than species in nature that are large-sized lianas.
Okay.
Lianas.
Okay. Interesting. And another question in Pilostylis, what's left from the radicle? Let's say from the root apical meristem, you mentioned it doesn't develop, but there is something, or it's aborted, or what's happening there?
Yes, so if there is a haustorium, just the one that you know from parasitic plants, but it's extremely reduced to a few cells that are more similar to the hyphae that Nilima's showed, Nilima's presentation from yesterday.
Yes.
Small cells that kind of enter the vasculature from the host. However, one of our students, the same person that was doing the shoot apical meristem analysis, is currently targeting all of those genes putatively involved in root development.
Okay.
But we expect also a large reduction.
All right. Yeah, but very, very interesting. Thank you very much.
Thank you.
Time for a very, very short question, a very short answer from Berenice Garcia. Berenice, go ahead. Go ahead, Berenice. Could you hear me?
Muted.
Okay. Okay, so there are two questions in the chat, Natalia. If you can answer directly in the chat, I will appreciate that.
Absolutely. Thank you so much.
Our next speaker is Jalian Ding, and he's coming from the Duke University in UK, and the talk is RNA structure, a hidden regulator in vivo. Go ahead, please.
Okay, can you see the screen? Can you see it clearly? Oh, okay.
Yeah,
Yes.
Should I start? Okay. Thank you very much for organizer to actually giving me the opportunity to talk about the research in my group. So today I'm going to talk about RNA structure functionality in plant system, how it actually regulated the gene expression. So if actually in the central dogma, RNA is actually in the central position, actually from the DNA to protein. But however, for a very long time in the textbook, you always can see all these mRNA is actually remain linear line, which is seems like it's completely single stranded. So why it actually always draw like a single stranded? But interesting, in the past like several decades, when people drop into test tube, into any different lengths of RNA or different sequence combination put into test tube, then they detect the RNA shape, the structure of it. You always can actually see certain structure like a stem loop. So sometimes it's forming very complicated structure like a pseudoknot or triplex or G-quadruplex. So why actually all these textbooks still remain like a draw messenger RNA as actually linear line instead of joining into a certain structure format? The most reason is because all these structure analysis is actually happening inside the in vitro condition. So if you don't know what the structure looks like in the living cells, so you couldn't really know, okay, what kind of shape really actually into their biological function. So pretty much about 6-7 years ago, several groups together's effort to try to changing the whole in vitro study, transform in vitro study to the in vivo study. So kind of develop a lot of different methods which try to profile the RNA secondary structure in the living cell. So what basically the transformative method try to do is actually using a kind of a chemical called dimersa sulfate. So which actually can penetrate it into the cell and actually modify the single-stranded nucleotide like A and C. So what it do is it actually attached a methyl group on the Watson-Crick phase of the N1A position and N3C position. So when these A or C is single-stranded, these methyl group will actually attach to it. Okay, once it attached to it, when we detect through the reverse transcription, because the methyl group is at the Watson-Crick phase, so the reverse transcriptase will stalling at a position which got modified. So that means if you detect a more reverse transcription story, that means the nucleotide is actually have a more modification of these dimethyl sulfate method group. So in that way, we can generate something we call a chemical reactivity. If you have a high chemical reactivity, means more reverse transcription story, that means this nucleotide is tended to be high probability as a single-stranded. Oppositely, is if you actually have less storing of reverse transcription, then this nucleotide are likely to have a less modification. So actually the probability of a double-stranded will be higher. So recent years, there's actually new method actually using longer reverse transcriptase to introduce the mutation instead of stalling to detect this kind of modification. So the other chemical, which actually synthesis from Eric Koo and Howard John's group, is actually shape a chemical compound called NAI. So what it do is quite similar, but instead of actually react on the nucleotide base, it actually reacts on the 2' hydroxyl group. So when the RNA is actually single-stranded, so this chemical will modify the acylation, the 2' hydroxyl group, leave a block on the RNA, so it also can be captured by the reverse transcription storing. So based on these kind of a chemical probing method, which cooperates with deep sequencing library, we will be able to actually probing the in vivo RNA structure at a genome-wide scale. So now we actually have all these really powerful tools. So then we kind of go back to ask what's really going on, or how we kind of rewrite the textbook. So the first thing is actually we look into is actually during the RNA maturation, the splicing process. So when the POL2 generated the nascent an RNA, you can see with the exon-intron, where you see the textbook, it's actually drawn as a linear line. It's like completely single-strand RNA. Is that true? So we kind of put that into one question of that. So the second actually related to the RNA biology process is actually RNA degradation. The most famous major RNA degradation pathway is microRNA mediated RNA degradation, where you see on the textbook all these microRNA target messenger RNA is actually drawn as a linear line as well. It's like it's completely single-stranded to wait for the microRNA to base parentase. Okay. The other process of RNA biology process is translation. So where you see the ribosome going through the messenger RNA, all these messenger RNA draw as a single line. It's like it's completely single-stranded, rather the ribosome going through it. So what we actually want to see is all these processes, all these RNA, whether or not they really look inside in the living cell, they actually still remain as what they draw, it is on the textbook, or actually they are different shape. So in the past 6-7 years, my group actually focused on the plant system using a Arabidopsis and model plant species, try to study all these functions relevance. So we also actually expanding all these technology towards brassica, rice, wheat. So today I'm going to focus on all the study we have been done in the Arabidopsis thaliana. So the first question I was trying to talk about is actually how does a spliceome recognize the true splice site? So when the POL2 generated the nascent RNA, when you're looking through crossing the exon-intron, you can find all these actual nucleotide motif like a GU, AGU, crossing over the whole nascent RNA, okay? So then the question coming through is why certain GU position will be recognized by spliceome, and some nearby GU motif, like a sequence motif, or later on in the intron, these GU motif never be recognized. And how actually spliceome decided to choose this one instead of the other ones. Okay, so then we hypothesize these nascent RNA should actually have a certain RNA structure feature allow the spliceome to actually recognize it. So to answer this question, we designed an experimental called NUC shape structure seq. What we do is we chemical treat the Arabidopsis thaliana seedling. Okay, all these single-stranded sites will be modified by the chemical. So then we enrich the nuclear to actually enrich the pre-mRNA proportion, so all the nascent RNA as enriched as possible. So generate a deep sequencing on the pre-mRNA RNA structure. So then we actually basically try to do this kind of metagene analysis. So what do we do? Is because we generate a deep sequencing library, RNA structure library on the pre-mRNA. So we separated these pre-mRNA into two different groups. One group is later, when it becomes mature RNA, this intron will be successfully spliced out. The other pre-mRNA group is actually this intron maintained, okay, in the later stage. So actually divided by different fate afterwards, okay? So then we basically just try to pull out the shape reactivity or the chemical reactivity to actually see how the difference between these two types of group of pre-mRNA. So very interestingly, when you actually see the shape reactivity, which means for high shape reactivity, more single-stranded, if lower, it's more double-stranded. So the interesting feature we observed, very limited nucleotide, is actually 2 nucleotides upstream of a 5' splice site. So if the last 2 nucleotides in the exon before the 5' splice site is a single-stranded, so it's always associated with a splice event. And also the other feature we saw is the single-strandedness in the branching point. So if it's always single-stranded, it's always associated with the splice event. In opposite, all these pre-mRNA later won't go in through the splicing event. Instead of actually have the intron retention, well, don't have this kind of a strong single-strandedness of 2 dinucleotide upstream of a 5' splice site, and also the branching point. So based on that, we actually want to see whether or not if alternative splicing has a similar feature. So we group a kind of alternative splicing event on the site which you have this site as a major 5' splice site. The other site is a minor. If you can see, if this position is actually the major, you can see the dinucleotide single-strandedness actually upstream of 5' splice site. Okay, so if you actually have the other site as the major one, you can see these 2 dinucleotide single-stranded upstream 5' splice site come up. But in the minor one, you don't have that. Okay, so that means this dinucleotide single-stranded upstream of 5' splice site is also kind of associated with alternative splicing. So then we kind of see this kind of association. We kind of wondering, okay, really, how these 2 nucleotide single-stranded really regulate the fate of splicing? So what we do is we try to validate it. So we took an individual cassette of a pre-mRNA, like this kind of example. You can see is there's a 2 nucleotide single-stranded actually upstream 5' splice site, and we transform this whole cassette into tobacco. And what we can see is actually successfully spliced, which agree with our metagene analysis. So if you have a single-stranded upstream of 5' splice sites, you will actually have a successfully spliced event, okay? So then what we do is actually because during the crossing, the exon intron had a U1 siRNA binding site, which is 3 nucleotides in the exon, 6 nucleotides in the intron. This 9 nucleotide is a U1 siRNA binding site. So try not to disrupt these nucleotides. We actually insert a sequence which can form a very strong stem to kind of block the base pairing, or the actually forming the base pairing with all these 9 nucleotide U1 siRNA binding sites. Okay, so once we actually inserted this kind of sequence, then this splicing event was abortion. So no splicing. Okay, so instead of changing this sequence, we're changing the opposite, try to changing the structure feature. Okay, so what we did, I still remember the -1, -2 position upstream of 5' splice sites. If it's single-stranded, it will be spliced. So we disrupt the base pairing of the partner nucleotide. Once we change into GGAA, the splicing coming back. Okay, so then we thought, okay, how about the other position crossing this whole stem? When another other position single-stranded will actually also changing. No matter where we actually changing these base pairing, It's only the 2 nucleotide upstream of a splice, 5' splice site is actually can rescue the splicing. The other side couldn't actually restore the splicing event. So we repeated this into different actually precursor or these pre-mRNA. We can actually see similar events. So this is something which is super shocking to us, like you changing the structure, you can changing the splicing fate. Okay, now we go back to the textbook, these kind of line again. At least we can know is if you have these 2 nucleotide upstream 5' splice site as single-stranded, it's likely to be recognized by spliceome. But if it's blocked as a base pairing, it's likely won't be recognized as actually the splicing site. So now we can update a little bit of the textbook towards the pre-mRNA structure. So now we go back to actually the microRNA mediate RNA degradation pathway. So quite a lot of people actually working on the microRNA mediate RNA degradation. So almost everyone think is actually the target site needed to remain actually single-stranded to be able to base pairing with microRNA. This is what actually people keep in mind how it should look like. So this 21 nucleotide will actually really inside the cell is single-stranded, we don't know. But previously, if using David Bartel's target founder, you're always using the complementarity to actually predict whether or not this site will be the target site. Okay, the rule is actually based on how many mismatch between these microRNA and their target site. If it's under 4 penalty score, it's supposed to be the target site. But however, sometimes when you see perfect complementarity, it's still not going through undergoing the cleavage. So why? So almost all the 70% of these kind of prediction seems don't have the cleavage really happen. So it's like a false positive. So then we had a hypothesis is whether or not this actually targeted messenger RNA had a certain structure feature which tried to control this microRNA mediate RNA degradation process. So what we did is we actually generated another type of structure library called a cap structure seq. What we do is we using a Arabidopsis seedling, we do the chemical probing, so all the single-stranded nucleotide will be actually modified by the shape. Okay, so among all the RNA population, there's a degradation product, also the intact RNA. We get rid of all these degraded RNA through different enrichment. So we actually only capture the intact RNA, so before the cleavage happened. So if you think about a structure as a cause and a cleavage is actually an outcome, then you will need an intact RNA, which before the cleavage happened. So we generate a deep sequencing library, RNA structure, on the intact RNA. So then the first question, obviously, is whether or not the target site is actually open, completely spatially accessible to the microRNA. So also whether or not it's actually forming into certain structure. The first thing we thought is actually not spatially accessible. So actually almost all the target sites are forming certain structure into a double strandedness instead of completely allow 21 nucleotide to be completely single stranded. So now you actually think about it, it's actually quite makes sense. If you have an RNA region above like 21 nucleotide, completely single stranded, then the other endonucleus will be easily to actually recognize it and cleave it out. So maybe forming certain structure crossing the target site is a way to protect unspecific endonucleus cleavage. So when we saw this target site actually is forming certain structure, then the next question is actually how it actually really kind of contributed to the microRNA cleavage. So because it's forming into certain structure, so then we'll be able to using in vivo RNA structure information to generate an energy landscape of actually whole reaction of a microRNA mediated cleavage. So the first step is where you have a target site forming certain base pairing, then you need to overcome energy barrier to be able to allow this target site to be completely open to base pairing with your microRNA. So you have a first energy barrier to actually overcome. Then later on, you're forming RNA duplex. Then the enzyme catalytic cleavage actually energy will overcome to cleave the RNA into 2 pieces. Okay. So then, based on this energy landscape, you're gonna have 2 scenarios. One scenario is this first energy barrier will be the speed limiting things. So how easy to open up to the single-stranded target site is actually determining the whole reaction when or not it will go through. Okay. The other scenario is actually the cutting energy barrier. So how easy the enzyme cleavage happening, so the reaction will go. Okay. So if this is a speed limiting step, then that means we can actually calculate this delta G, so the energy barrier, then with the cleavage efficiency. So we did a correlation with cleavage efficiency with this energy barrier. Okay. So what we can find is a significantly anti-correlation with cleavage efficiency, which means if this delta G open barrier, so more easy to open the target site structure, the more easy to be cleaved. Okay. But in contrast, if we take the energy barrier for the enzyme cleavage activity, the energy barrier, you can see there's no significant correlation with cleavage efficiency. So in the plant system, we think it's actually the first delta G, the energy barrier, is the speed limiting step. So this is actually quite agree with organelle pull down experiment. You normally don't get a Y type organelle pull down with actually the messenger RNA species. You have to have a cleavage mutation to pull down the messenger RNA. The main reason we think is once argo drive the microRNA binding to the target site, so once it's binding, the cleavage will happen. So it's very fast kinetics to follow this whole reaction. So then, of course, we know the target site. The next thing we want to know is actually how franking sequence structure really contribute to this process. So again, we did a meta-G analysis. So because the microRNA target site had a different size because different size of microRNA. So we aligned the 5 end of microRNA's target site. Then we aligned the other 3 end of microRNA target site to looking for the 3 end of franking sequence. And also 5 end is looking for 5 end of franking sequence. So what we can see is we separate the 2 groups. One group is later on won't be going through the cleavage, okay, non-cleavage. The other group is actually it will cleave. So now come up, the only difference we can detect is 2 nucleotide downstream of the target site. Again, dinucleotide single-stranded downstream of the target site. Control seems associated with the cleavage event. Okay, so then we thought, okay, how we validate it? So we actually put a microRNA 156 target site into the cassette, expression cassette. So after that target site, we put a stem loop structure. Then between the target site and the stem loop, we put a slippery sequence we call target adjacent mediate motif. So single-strandedness 2 nucleotide between target site and the stem loop. Okay. We also have another quite a strong structure called the G-quadruplex after the target site and put the slippery 2 nucleotide between the target site and the G-quadruplex. Interesting happening is if the target site follow with the stem directly, you don't have cleavage. Okay. If you put the TAM, which is a 2 nucleotide single-stranded between the target site, downstream of target site, then cleavage happening. Okay, this is measure the RNA expression. So it's actually because of degradation, the expression level going down. So then this is G-quadruplex without a TAM, no cleavage. With TAM, cleavage happen. Okay, then because of that, we know, okay, this dinucleotide single-stranded nucleotide downstream of target site corresponding to the microRNA cleavage activity. So the question then is actually whether or not this time the dinucleotide single-stranded influence the cleavage or influence the binding. Okay, so then we collaborate with Peter Balderson's lab, try to perform the cleavage assay. So what we did is actually we put this kind of a structure without the TAM, with the TAM, with organelle, extracted organelle with the microRNA substrate inside. So we take the Y-type argo to perform the in vitro cleavage assay, which you can see once you have the TAM, the cleavage actually happening. Without a TAM, don't have a cleavage. Okay, So meanwhile, we collaborated with Alberto in Spain using his construct, which is a cleavage mutation, which only have a binding capability with organelle protein. We're using that to perform the in vivo binding assay, which we can find is with TAM, without a TAM, the binding didn't affect that much. So that means the TAM region is actually corresponding to the organelle cleavage. So now we go back to capture the whole process of actually the structure contribution to the microRNA mediated cleavage. Is actually, you can see at the very beginning, your target site needed to overcome an energy barrier to open as a single stranded to form base pairing duplex with the microRNA. So if you have a downstream 2 nucleotide is a single strand, which we have a TAM kind of motif, then you are going through the cleavage. If you don't have TAM, it won't go into the cleavage. So now I want to actually show you some kind of evolution between the organelle and the Cas13. So if you can see the Cas13, after the target site, there's called a PFS motif, which is non-G motif. So if you're looking closely about the guard RNA, CRRNA, you can see the opposite nucleotide is actually C, corresponding to the downstream target site, okay? So if this is C, the other side is G, then we'll form CG base pairing. So that's why no cleavage will happen, okay? So, but if this is the other nucleotide CAU, once it couldn't form base pairing with C, the cleavage will happen, okay? So now you're looking to what we saw on the organelle 1 is actually downstream of target site. If it's actually single-stranded, then the cleavage will happen. If it's actually double-stranded, it will uncleave. Okay, so because we are really plant biologists, we want to see, okay, if this is truly super powerful, so whether or not we can actually change the real fate of these famous microRNA target gene. So the gene we visit is actually, I think Natalie just talked about AP2, APATL2, which is a flower development identity gene. I think Xuemei is famous for actually gene. So this actually AP2 is undergo microRNA 172 target. But however, it's actually don't have a strong cleavage inside of the microRNA target. So if you actually have this is a microRNA 172 target in the AP2. So when we see the structure, we can see downstream 2 nucleotide forming base pairing with upstream of the structure. So when we saw that maybe because this TAM region actually blocked as double-stranded, which causing the cleavage won't happen, okay? So what we do is instead of changing the TAM region, we're changing the base pairing partner. Just you think about it, it's a coding region of the AP2. So we only changing the synonymous mutation of this A nucleotide of the base pairing partner, okay? So if it's Y type, it's the same as this structure, nothing happening, so no cleavage. Once you're changing this A synonymous mutation to G, U, C, cleavage happening. Okay, so this is doing in the microRNA overexpression, 172 overexpression of Arabidopsis protoplast. Okay, then we want to see, okay, if we're moving to inside of the plant genetics, whether or not it's still working. So if you see, this is Columbia with beautiful Arabidopsis petal, white petal. So this is AP2 non-mutant, which don't have a petal. If you complement with the Y-type sequence, you got a petal back. If you complement just with a synonymous mutation, A to G, A to U, A to C, it didn't coming back. The microRNA, actually, AP2 was degraded in these lines. Okay, so that's why phenotype typical, you actually can see that. So this gives us really strong confidence how powerful RNA structure is. So you basically can see is actually structure can kind of changing the fate of a microRNA cleavage, and actually this will actually causing a strong phenotype changing. So I just gonna wrap that is actually microRNA cleavage regulation is if you first the target site forming certain structure, then you actually have a TAM. If you have a single-stranded downstream of a target site, you actually will go into cleavage. If not, it will actually block the cleavage. So now we go back, is actually the RNA degradation is actually have this kind of a feature of the structure there. Okay, so did I overtime that? I can just shorten it. So basically, I can just shorten it into that space. So basically, this is pretty much how much we actually changing. I didn't talk too much about the other part on the translation and the stability. So basically, is we kind of starting knowing that RNA structure starting have a certain function inside the actually RNA biology process, which possibly influence the splicing fate, influence RNA degradation, also the translation. And I think I'm running over time. So basically, I want to thank my whole group and also all the funding agency. Thank you.
Thank you very much, Glenn. We have time for a very
bit over.
Gerardo, please open your mic.
Yeah.
Gerardo, please open your mic yourself.
Yeah, can you hear me now?
Yeah.
Yeah.
Yeah, okay. Thank you. Very interesting talk. So I was wondering if the presence or absence of this time can also trigger, for instance, generation of secondary small RNAs triggering post-transcription and gene silencing, for instance. I don't know if you have a look at that.
So what we actually currently do is actually, because we know the rule, right? So if you want to actually silence a gene more effectively, you can using this structure guide. So because we have, for example, we have in vivo RNA structome of Arabidopsis different virus. So we're actually doing that now. So we're basically using our application more detailed rule towards how structure information can guide the silencing efficiency to actually improve the microRNA cleavage. Yeah.
Okay, sorry for the other two people that want to ask, because we don't have time now, but maybe you can do the questions in the chat and
yeah.
the answers there. Thank you very much.
Sorry for overrun the time. Thank you. Thank
you for this Congress. Thank you.
Thank you.
Hello. Our next speaker is Luis Herrera Estrella from Langevio, Sinvestab, Mexico, and he's going to talk about genome accessibility dynamics in response to phosphate limitation is controlled by the PHR1 family of transcription factors in Arabidopsis. Go ahead, Luis, please share your screen.
Okay, thank you very much for the opportunity to present some of the results of my research group in this very interesting Congress. So I'm going to talk about the effect of phosphate starvation on changes in chromatin accessibility in response to phosphate starvation. And I'll be talking mainly about the work of Dr. Lenin John and Dr. Carlos Barragan. So I mentioned them before I forget to give the credits to the people who actually did the work. So I just want to first mention the problem of phosphate. Phosphate is a limited nutrient in 70% of the world arable land. And this is mainly due to the chemical properties of phosphate, which very rapidly reacts with cations present in the soil particles, either in acidic or alkaline soils. And this makes that the phosphate gets fixed and unavailable for plant uptake. So when roots grow in the soil, they create a depletion zone when there is no more phosphate available for uptake by the root. Therefore, the plant has to respond to adapt to low phosphate soils by changing the growth of the root and also by changing its metabolic pathways to reduce the use of phosphorus. So phosphate is a very inefficient process because of the phosphate you add into the soil. Perhaps only 20 to 30% is efficiently used by the crop, and the rest is lost due to fixation and microbial activity. So because of that, we have to apply an excess of fertilizer, phosphate fertilizer that causes serious ecological problems and also poses a great danger in the life of the phosphoric rock reserves from which all the phosphate is extracted. So to come with phosphate limitation, plants activate mechanisms that facilitate phosphate uptake, scavenging from the soil, and optimizes the use of internal phosphate resources to secure plant growth and reproduction. So this involves the activation of genes that encode high affinity transporters, that encode acid phosphatases that are secreted into the rhizosphere to hydrolyze organic compounds containing phosphate to release the phosphate for plant uptake, nucleases. There is a whole metabolic reprogramming of the plant to reduce the need of phosphate, to concentrate phosphate for essential reactions. The plant also releases organic acids and other exudates that facilitate the solubilization of phosphate complexes in the soil, and changes in the architecture of the root system to increase the capacity of the plant to explore the upper layers of the soil where phosphate accumulates. So there are two main responses that plants activate. One is a systemic response that is responsible for the metabolic adaptation of the plant and activating all the genes involved in phosphate uptake and scavenging, and a local response which is responsible mainly for the changes in root architecture, like root hair elongation, the formation of additional lateral roots, and so on. This response is mediated by the activation and repression of many genes. So there are hundreds and in some cases thousands of genes which are either activated or repressed in response to low phosphate availability. And it has been reported already by the group of Javier Pasares and Ping Wu in China that this process is regulated by a central transcription factor, which is called PHR1. PHR1 is negatively regulated by SPX1, which is a repressor that binds to PHR1, blocks the DNA binding domain of PHR1, and prevents PHR1 from binding to its cognate target sites in the promoter of genes that are activated by low phosphate. This interaction is mediated by inositol polyphosphate. So when the plant is grown in optimal conditions of phosphate, the concentration of inositol polyphosphate increases, and it mediates the binding of SPX to PHR1 to maintain it inactive. Under low PI conditions, the level of inositol polyphosphate drops, then PHR1 is released from SPX1, which is degraded by the proteasome, and then PHR1 is free to bind to its cognate sites in the DNA, in the promoter of genes that are responsive to low phosphate. PHR1 activates only a subset of the genes that are involved in the low phosphate response. So there has been estimations that PHR1 directly binds to the promoter of about 300 genes. And the remaining of these 300 genes are activated probably by transcription factors that are direct targets of PHR1, and in turn activate the expression in a second wave of gene expression activation in response to low phosphate. This response is being studied mainly at the molecular level, at the transcriptional level, in terms of what is the role of these transcription factors. PHR1 is member of a small subfamily of mid transcription factors composed in Arabidopsis by 5 members that all participate in the activation of the low phosphate response. And PHR1 is the most important one because if you mutate PHR1, the response is drastically reduced. But we have observed that when you grow plants on the low phosphate, the next generations of the plant are better suited to respond and grow better under low pI conditions, suggesting that there are some epigenetic mechanisms involved in this response that allow the plant to have a better response when they have exposed once or twice to this type of stress. So as you know, epigenetic mechanisms evolve different levels from DNA methylation, histone modification, and chromatin remodeling. So the first thing we did is Lenin Young studied the changes in global DNA methylation in the Arabidopsis genome in response to low phosphate. So we use whole genome bisulfite sequencing, and I take this opportunity to just mention that we have reported a paper to determine the methylome of Utricularia giva, which is a small carnivorous plant, in which we show that you can use also directly PacBio sequencing to determine the level of 5-methylcytosines in the genome of this plant, and the pattern of distribution of methylated cytosines is very similar to that produced by the bisulfite sequencing technology. So if you have data from PacBio, you can directly use it to determine the content and distribution of 5-methylcytosine in your favorite organism. So using this bisulfite sequencing, we determine the effect of low phosphate on methylation, and we determine differentially methylated cytosines and differentially methylated regions in the genome of Arabidopsis. And I just want to mention a few things about this, that the methylation is dynamic. It changes from, depending upon the time of exposure to the stress, from 7 days to 16 days of exposure to low phosphate. You can see that there is an increase, for instance, in CG methylation in the shoot, and that the response in methylation between organs, the shoot and the root, is different. So for instance, here, CHH is the CHH methylation context is the most important in the shoot at 7 days after exposure to low phosphate, whereas in the root, you have 36% of CHH and a similar amount of methylation in the CG context, showing that the methylation changes induced by low phosphate different between organs. And these differentially methylated cytosines are present in different parts of the genome. They are preferentially observed in the gene body, including the 5', the coding sequence, and the 3', and in transposable elements that are located close to transcriptional units. Among the different methylation contexts, CG, CHG, and CHH, we found that the CG context is the most important and the most prevalent, that are differentially altered upon exposure to low phosphate in the gene body regions of the genome. As I mentioned before, the differentially methylated regions, this is not single cytosines, but regions that windows of 50 bases that contain differentially methylated bases compared to the untreated control. You can see that there are very important differences in terms of which regions are hypermethylated and which regions are hypomethylated between the shoot and the root, so suggesting that the responses of the two organs are different, although many of the genes that are induced in the root are also induced in the shoot, but not all the genes that are induced in the root are also induced in the shoot, and vice versa. So these methylated regions are mainly associated with gene body regions of the genome, but also there is a significant amount of differentially methylated regions associated with transposable elements located in the vicinity of the transcription start site of genes. When we did an analysis of the type of genes that were differentially methylated in response to phosphate, we, perhaps as expected, we found that genes involved, important genes that involve are involved in the response to low phosphate, such as those involving the synthesis of galactolipids that replace phospholipids in the membrane of the cell to allow the release of phosphate from phospholipids, the SPX domain proteins, acid phosphatases, all these genes undergo differential methylation. Some are hypomethylated and have higher expression, others are hypermethylated and have reduced expression. Among these genes, we found several patterns of methylation that are related to changes in transcription levels. So for instance, in the case of SPS2, what we found is that there are differences within the transcribed region of the gene, and when this region is hypomethylated, we have a higher level of expression. The most typical part or the most logic way of explaining the impact of methylation is when you have hypomethylation in the regulatory region of the gene and you get higher expression. That was the case, for instance, of this microRNA that is also activated by low phosphate. And we also found genes that were activated when transposable elements were hypermethylated. So this transposable element is located near to the transcription start site, and when it gets methylated, the expression of the gene is increased. So as I mentioned before, we have different levels of regulating epigenetic processes. So DNA methylation and histone modifications finally result in changes in chromatin accessibility. So Carlos Barragan decided to study the impact of low phosphate on chromatin remodeling. And for this, we use the transposase accessible chromatin assay, the ATAC-seq, which is a very versatile system in which a hyperactive transposase binds to regions in which the DNA is exposed and inserts adapter that allow to sequence the regions that are in DNA sequence that are located in regions of open chromatin. So when you have chromatin, the transposase produces fragments that can be PCR amplified, and you can get the sequences of the regions that have open chromatin, and it will not do so in regions where you have closed chromatin. This technology is quite friendly because you need few cells or few nuclei to do the process, and the preparation and time to produce the sequencing libraries is much faster than other methods that are commonly used to determine changes in chromatin accessibility. So what we found is that upon exposure to low phosphate, we find differentially accessible regions. The number of differentially accessible regions increases in the genome of Arabidopsis. You can see here a distribution of the red bars are increase in chromatin accessibility, and the blue one are decrease in chromatin accessibility. The blue ones are not, most of them are not statistically significant, whereas the increase in accessibility, many of them are statistically significant. And we can see here, for instance, one of the genes that is responsive to low phosphate, phospholipase CETA2, which is involved in the remodeling of lipids in the membrane in response to low phosphate, gains chromatin accessibility in its promoter region upon exposure to low phosphate, and that correlates with the activation, the transcriptional activation of this gene. So we found that there are differentially accessible regions that win accessibility and some regions that lose accessibility. Most of them have higher accessibility, and a minor proportion have lower accessibility. And interestingly, many of these regions of higher or lower accessibility are located in the 5' regulatory region of genes, although there are also an important number of these regions that are in intergenic regions, which we cannot assign a function because they could be associated with enhancers that act as a distance. So to understand the effect of these regions that have higher or lower chromatin accessibility, we associated these regions with the closest gene to see what was the effect on the transcription of genes. So when we associated these DARs, or differentially accessible regions, with genes, and we did a co-enrichment of these regions, these DAR-associated genes, we found that there were categories that were involved in gene expression, cellular metabolic processes, but nothing really related with phosphate metabolism or phosphate starvation, which was a little bit surprising, but not so much, because from the thousands of genes that are activated by low phosphate, perhaps only 1/4 or 1/5 are directly associated with the low phosphate rescue response. So we did RNA sequence analysis of the same plants for which we did the ATAC-seq analysis, and we found that, as expected, there are many genes that are induced and many genes that are repressed, and most of the typical genes that are induced by low phosphate are also activated under our experimental conditions. So then we did an association of these DARs with the differentially expressed genes, and we found that there were 537 genes that were associated with up or down DARs. And when we did a co-enrichment category of only these DARs that are associated with differentially expressed genes, then we found that these are enriched in the cellular response to phosphate starvation. If we associate the UP-DARs with UP-DEX, these 247 genes, so there are about 1/4 of the genes that are upregulated in response to low phosphate are associated with regions that gain chromatin accessibility. They are, again, enriched in categories related to responses to stress, and in particular, responses to phosphate starvation. So one of the questions we had is, because PHR1 plays such a major role in the activation of these genes, whether the changes in chromatin accessibility were dependent or not on PHR1. So for that, we did an analysis of a PHR1, PHR2 mutant. This double mutant has a very significantly reduced response to low phosphate. As you can see here, from thousands of genes that are induced or repressed, there are only a few hundred that remain to be active in this double mutant. Perhaps these genes are activated by other members of this family. And most of the genes that are expressed under our conditions in the root of these plants that are direct targets of PHR1 are no longer expressed or have lower expression in this double mutant. When we did the chromatin accessibility in the double mutant, what we observed is that the changes in accessibility observed in the wild type are no longer observed in the double mutant, and the number of regions that are differentially accessible is significantly reduced. So the number of UP-DARs regions that increase accessibility comparing the wild type with the double mutant decreases significantly as the number of genes that are differentially regulated. So this suggests that not only PHR1 is involved in the activation of the expression of these genes, but also in modulating the status of chromatin accessibility in the Arabidopsis genome. So then we did an analysis to see what was the change in chromatin accessibility of the genes that are direct targets of PHR1, of about 230 genes that are expressed in the roots of Arabidopsis. About 90 genes undergo changes in chromatin and accessibility. 156, which are not direct targets of PHR1, also undergo changes in chromatin accessibility. These changes mainly occur on the wild type, and very few have the same chromatin changes in the double mutant, suggesting that in fact PHR1 and PHR1 are responsible of activating these changes in chromatin accessibility. When we analyze the Go categories enriched in these direct targets that undergo chromatin accessibility changes, we find that they are all very enriched in cellular responses to phosphate, suggesting that the direct targets of PHR1 that increase chromatin accessibility are central components of the cellular response to phosphate starvation. So we found that among we found 16 genes that were partially dependent on PHR1, PHR2, because they were activated in the wild type, but also in the double mutant. But these genes were activated in double mutant to a much lower extent than in the wild type. Those genes are related with central reactions involving the low phosphate starvation response. Among the 74 targets of PHR1, there are many canonic genes that are activated by PHR1. And there are also a number among these PHR1 target genes that undergo differential accessibility of chromatin, are many transcription factors that probably are involved in a second wave of activating gene expression, and that probably are responsible also for remodeling the chromatin of the genes that undergo again in chromatin accessibility, but that are not PHR1 direct targets. So this is just a comparison of the average of changes in chromatin accessibility in the genes that are responsive to low phosphate and gain chromatin accessibility. You can see that there are increases in chromatin accessibility in the 5' and 3' regions of the genes. And this accessibility is completely lost in the 74 genes, which are only activated in the wild type, but not in the double mutant. Interestingly, when we analyze these genes that are activated still partially in the double mutant, we find that they still have a partial response, but that they also have this chromatin modification in plants that are grown under PI sufficient conditions, suggesting that this might be actually being more highly expressed on their optimal PI conditions. So how does PHR1 activates changes in chromatin accessibility? Some people have reported the discovery of pioneer transcription factor. Usually what thinks that transcription factors bind to accessible chromatin regions to recruit the transcriptional machinery and start transcription. But there are some transcription factors that can actually recognize its cognate binding site in closed chromatin, recruit chromatin remodelers, open the chromatin, and then recruit the basal transcriptional machinery to activate transcription. So what we found is that in many cases of the genes that are activated by low phosphate, and they are direct targets of PHR1, in seedlings that have optimal PI conditions, there is absolutely no signal of open chromatin. When you expose the seedlings to low phosphate, then you have a peak of chromatin accessibility. And when you have the double mutant, there is little or no change in chromatin accessibility. Then this suggests that PHR1 binds to closed chromatin structure, and that's the way it activates transcription. So that's about what I just wanted to tell you. I just want to say that under low phosphate, there are some genes that are actually activated by PHR1 at optimal conditions at low levels. And when there is low phosphate, PHR1 binds to closed chromatin structures, open chromatin, and activate transcription. Thank you very much.
Thank you very much, Luis, for accepting this invitation. We have time for questions. If someone have a question, just raise your hand. Maria, open your mic yourself, please.
Is it Maria or is it Mario?
Mario, sorry.
Okay.
Oh, sorry.
Luis, really nice talk. I just wanted to ask you, how do you envision that the open chromatin or the accessibility of chromatin is established if it's dependent on RDDM, or if it's something that relies directly on chromatin modification but that do not use small RNAs?
We really don't know. What we have found is that there are many subunits of different chromatin remodeling machinery that are actually, their expression is dependent upon PHR1 on their optimal conditions. And there are some protein-protein interaction directly with PHR1. So what we think is that maybe PHR1 directly recruits the remodeling machinery.
Thank you, very interesting.
Yeah.
Joseph, short question, please.
Yes, hello, Luis and everybody. My question is related, it's very interesting material. You presented very interesting analysis, but I wonder if you check if there is a correlation between DARs and the genes expressed which are active in lateral root formation, because as root system architecture is changed under low phosphate, we can expect that, right? But it would be interesting, I don't know if you check that.
We haven't finished the analysis and we haven't examined that part, but we don't know whether these genes are going to be, because we center our analysis on PHR1 direct targets, probably the genes that are involved in changes in root architecture are independent of PHR1, because the PHR1 mutant still responds in changes in root architecture. So, but we haven't checked if those also change chromatin accessibility. That's a very interesting point, Joseph.
Okay, okay, thank you very much.
Very much again for your very, very nice talk.
Thank you, Luis. So we'll go to the next talk that will be presented by Jim Hasselhoff. He's from the University of Cambridge, UK, and we appreciate that you accepted this invitation, Jim. So go ahead.
Thanks very much for the invitation. And I'd like to start just to describe my talk as being a general approach of looking at engineering principles and how they might be applied in plant systems. And I guess the first question is, why does one think that's important? And so this slide shows sort of historical view of plant improvement with on the left-hand side here, the conventional plant breeding, where we have the selection of variation to create improved lines. In 1983, we saw the publication of the first transgenesis papers, where individual genes were introduced into plants and could be used to improve germplasm. But in the last 10 years or so, we've seen the increase in capacity of engineering, the introduction of gene editing techniques, and also reprogramming of existing gene circuits to create changes in morphology, changes in multigene traits. And the key aspect over the last 10 years has been that these new approaches allow for the first time really rational approaches to the engineering of new forms and traits inside plant systems in a way that outstrips the earlier technologies. And so arguably we're now faced as biologists with these complex systems that these kind of approaches have been enabled by increased information and knowledge about the systems, but now faced with dealing with the kind of complexity that's required to engineer these things in a coherent fashion. So we have, for example, the DNA systems that underpin plant form and behavior shown here in yellow, where we've got these systems which involve regulatory networks, which control properties of metabolism and structural genes underneath these hierarchical systems. And of course, these inform the properties of individual cells inside tissues, inside the organism. So in order to get traits at the whole plant level, one needs to use DNA code. That's our tool. That's the way we can intervene inside plant systems. But we need this code to be able to interact both with the multiplicity of genes and other elements underneath these hierarchical systems, but also then play out in terms of the cellular interactions that drive the processes. So we're dealing with what is formally a complex system, and increasingly we need to take more direct and approaches to manipulating these systems. So in the last 15 years or so, the synthetic biology field has taken on board this challenge of addressing complexity at the DNA level. So this diagram shows in a hierarchical fashion the ability of taking raw code, identifying encoded functions to create modular elements, and that these modular elements can then be assembled, often with modular and reusable fashion, to create devices and circuits and ultimately systems that play out in the multicellular organism. And the benefit of taking these lower level systems and modularizing their process is both technical, which allows faster assembly and more reliable assembly, but it also encompasses some of the benefits that engineers have adopted and taken for granted over the many years in other engineered systems, where you get the benefits of abstraction and decoupling of fabrication from design in these in increasingly complex systems. So this process is common with many engineering systems, where you have underpinning elements where these can lead to applications, and these might be, in the case of electronics or biology, quite similar challenges in a way, different levels of complexity, different levels of elements, but nevertheless, this challenge of bringing together basic knowledge, which you might represent by academia or the kind of publicly funded research activities, and this basic knowledge being harnessed in terms of the intermediate middleware, you would call it, in the electronics and software industries, where you've got standardized elements. They might be electronic components, or they might be modular DNA components, and having either model systems or software glue that ties these things together to create the kind of end products that we want. And this is a very broad way of looking at things, of course, but there's also, it's not just a conceptual challenge, it's also a human challenge, a social challenge of bringing together what in biology would be driven from the natural sciences side of things, and natural scientists looking at existing biological systems on one hand, and trying to couple that with an engineering approach, which aims, rather than to glory in the complexity of a biological system, aims to simplify and reuse elements. And so there's a set of cultural links that need to be made between engineering and biology, as well as practical tool sharing between the two. So this linkage between, or parallel approach between electronics and biology, we've taken quite literally in a way of trying to glue fields together more closely. And so, for example, you can think of cells, biological cells, in an abstract fashion as taking on board inputs, be they signals, be they physical strains or stresses, and also producing outputs, which might be, for example, gene expression or changes in metabolism and cell communication. So you can think of these cells as being effectively handling information and creating changes. And of course, in an electronic context, you have similar kinds of processes where the inputs can be the input from sensors or switches, and outputs, of course, can be actuators or displays. And so what we've tried to do is to take these two approaches and effectively glue them together in order to create an environment which we call Biomaker, which is a way of bringing together engineers and biologists in a very, you might say, intimate way, where they have the opportunity to co-create, where it's always been quite difficult to have biologists and engineers working on the same problem because there are differences in approach, differences in language, differences in knowledge bases. And trying to bring those together, we think, is a worthwhile and important challenge. So what we've done is to take together some simple hardware. So this image shows commercially available electronics board. It has effectively a microcontroller, a brain if you like, a series of input and output devices, and is relatively cheap and can be used as a way of introducing biologists to more formal approaches in programming. As well as the actual hardware, we also use a graphical coding interface, which is called XOD. And we have training materials that we've developed here, which provide step-by-step instructions on how people who have no experience in programming can come to this and make systems that involve information transfer and output and those kind of control systems. And I won't go into details, but the paradigm that's used here is a data flow paradigm where you have these nodes, these boxes here, which have input and output nodes, if you like, on each of those boxes. And those ports deliver information across the system in a directed way. And you simply wire up these nodes to have information flow across the system, where each one of these nodes might correspond to a piece of hardware or an output device or something like that. So we have these systems, and the beauty of this is that you don't require any prior knowledge of coding. A biologist can pick this up effectively overnight and start to use it to make useful things. And so that was important to us to try and get people very directly involved with these things as quickly as possible. And one example I show here is a piece of electronics, which is cheap. It's about $1 each. These little transistor-like devices is actually a highly integrated device, which is extremely useful for measuring temperature. And so one of these devices can be abstracted in this programming environment. And so this is effectively the logical equivalent of that device. And these devices can be wired up to the microcontroller, and it will produce a calibrated output, which is the temperature that it's seeing. And so it's quite useful, of course, in biological instruments and elements. So this simple process means that you can start to run workshops where students can come without any programming experience and start to build things directly within a few hours. And so we have here one of these programming environments where you've got the different bits of hardware lined up. You have here, these are actually students in Yulapa in a workshop that was run just before the COVID outbreak with Mario Arteaga. And we've also now have a Spanish translation care of Mario and his daughter and Steph Norwood. And so we have resources that allow one to bring biologists together to work on programming processes, to build bioinstrumentation. So you have this dual issue of both utility. You can make something that's useful in the field or the laboratory, but also gather people together in mixed teams. So biologists can handle this programming environment, but they can also bring themselves together with engineers and co-create different systems. And a couple of examples here. Here is a series of pieces of hardware, for example, that you can bring together to create a device. So you can effectively glue them together in a 3D printed housing and use these programming environments, all code-free, to create interfaces. And this particular device is a controlled temperature device. And you can see here the temperature being read out here. And this is Fernando Guzman Chavez is using this incubator to run a low-cost lamp reaction. And Fernando, who's about to move to UNAM as well, he's originally from Mexico, been working in Cambridge for several years, is also working on programmable cell-free biology to make the reagents that can be used to drive these reactions. So there's this whole ethos, if you like, of bringing together low-cost, accessible technologies which enable people to customize and build instrumentation, which is also useful at the research level as well. So here's several examples from Antonio Ruiz Gonzalez in the lab, who's been working with plant and soil-based sensors, and it shows some of the elements that he's been using to put together, which of course are all reusable as well. So they're described on a common website and are all accessible. So that's the sort of the engineering side of the bringing together biologists and engineers. But we also, there's the other side of it is, it's all very well to be able to manipulate electronics with software, but what about the plant systems themselves? And this slide shows some of the, I think, important reasons as to why engineering systems and engineering approaches are so important at this stage. One is that plants are our most important resource for sustainable bioproduction. agriculture is ubiquitous and cheap, and plants are clearly programmable. They are very plastic, effectively. And we don't really have time for the conventional plant breeding approaches, given the challenges that we're facing in terms of global warming and other kinds of impacts, environmental impacts, and the demand for plant-based materials. But of course, there's a problem here because plants are difficult. They're characterized by slow generation times, complex genomes. The developmental aspect of plant growth is also very difficult to access sometimes, and difficult to analyze at the cellular scale. So in our case, we've shifted towards Marchantia polymorpha as a target for these kind of, you might say, bioengineering approaches. Marchantia is a flat prostrate plant. It's a bryophyte, it's a liverwort, and it has many elements or features which make it, I think, ideal for this kind of work. So it has a modular structure. So the top surface of the plant has these simple modular repeating structures. You can see these air pore-like structures over the top of each of these modules. If you look at that in cross-section, you can see the air pore here, and it forms this chamber-like device, which is effectively the photosynthetic unit in these simple plants, where this repeats across the top surface of the plant. And you can see here these filamentous cell structures which are packed with high levels of chloroplast and photosynthetic machinery. So you have this simple architecture. One of the other features about Marchantia is that it has a very convenient life cycle. So it is split, it's haploid in the main part of the dominant part of the life cycle. It splits into female and male parts, and then undergoes the sexual phase with fertilization at this point. And you can see here female and male plants. The male plants liberate motile sperm, and they can be used for very simple crossing of the female plant. And then within a few weeks, you get the formation of these yellow sporangia as a product of the fertilization. And each one of these sporangia has several 100,000 spores on board. And these, of course, have undergone meiosis and ready for the next generation. The spores themselves are like about the size of a yeast cell, and these are prolific, so a single cross will produce millions of spores. And the spores can be stored, but then when germinated, they'll grow very quickly. So if this is at day 0, within a day and a half to the same scale, you see one of these spores has expanded and you have this spherical cell with differentiated chloroplasts. As time goes on, you've got now 2.5 days. You can see after this is one of the original spores which hasn't germinated. So you can see after 2.5 days, you've got this cell having undergone the first division, and so on. So you've got day 3, you see the formation of the first sort of apical basal differentiation to form this rhizoid or root hair-like structure. And as time goes on, you see the formation of this sort of thalloid-like structure as it starts to form together with additional cells down here making the rhizoid-like structure. So after a week on an agar plate, you have effectively something which is effectively the size of a microbial colony, but is a small organism. And for scale, we have here a pipette tip showing one of these organisms on a plate. So it opens up new ways of working with plant systems, which are very interesting for us. And the other aspect, we're not quite finished there, because the other aspect is that Marchantia has this vegetative cycle for reproduction, which is even faster than the sexual phase. So on the adult plants, you see the formation of these cup-like structures and the spontaneous production of these gemma or vegetative propagules from these systems. Here's one of these cup-like structures. And you can see here individual gemma stacked up inside the gemma cup, where they're produced from an embryogenic layer at the base of the cup. So they're clonal propagules, and they're produced by the hundreds from individual plants within a few weeks of planting. So there's a very short generation time. And the products have these very simple cellular architecture. So you can see here a single gemma here under a 5 or 10 times objective. And the individual cells you can make out here, these will then germinate and grow themselves where the developmental process is in the open. So you have a completely accessible system for perturbation. And this is the basis for what we've been now building or using to build modular DNA tools and cellular tools to examine this. So we've been developing or adopting common syntax for making DNA parts for this simple plant system. So here we have a generic view of a plant gene effectively carved up into the Golden Gate, MoClo, or loop assembly standards, where you've got these 4 base overhangs, which define boundaries between different formal portions of these modular gene parts. And so by sticking to this syntax, or just establishing it and sticking to it, we have a system that allows both rapid assembly and also, importantly, exchange of parts between groups. And so this is something which has been extremely useful in terms of promoting shared work in the field. And so with this comes the use of these common syntax modular parts for assembly. And so Golden Gate and MoClo are quite familiar to many of you, I imagine. The version that we use is slightly simpler in the sense that it uses a recursive assembly process. And this was a strategy developed by Bernardo Pollock and Fernand Federici. And it uses the common 2 type 2S enzymes, but rather than having a hierarchical series of vectors, it uses a recursive model. So you simply go back and forth between odd and even named receivers. And each time one of these type 2S assembly protocols takes place, you remove the existing enzyme and shift to the next enzyme, and you can build up constructs in a sequential fashion, which is shown here. So you start with these common level 0 parts, which can be used to make transcription units. These can then be strung together 4 at a time to make larger scale gene assemblies. And this approach has been facilitated by the speed with which you can work with Marcantia in terms of transformation as well. So this shows a shorthand view of the speed with which you can work with Marcantia. We're actually using germinating spores for transformation. And these are based on a series of innovations that have come out of several labs in Japan, in Kyoto and Kobe and elsewhere, and adopted widely among the Marcantia community. And so using germinating spores, you can get rapid transformation and selection within a few weeks of candidate transformants. What's more, you can also grow through the entire life cycle without the need for a glass house or any other larger structure, just simply in a growth room or growth cabinet. And these are these Sacco 2 microboxes that we use, which use a woven polyethylene filter at the top here. You can see this green strip here, which has this woven polyethylene, which retains water but allows good gas exchange. You can see the levels of gas exchange, which can be up to 80 volumes per day, which allows a healthy growth of the plants in indoor conditions. So using this combination of DNA assembly tools, techniques, and growth techniques, you can start to string together larger scale circuits quite quickly. This shows one example. This is a combined work from Susana Suargueto, Bernardo Pollock, and Mihail Delmans, showing that you can string together genes of choice. So each one of these corresponds to a gene with appropriate control sequences, with an antibiotic resistance marker, and in this case, 3 different fluorescent markers. And you can see this multispectral image of one of the products of this kind of transformation, showing different genes. We've got the Marcantia version of topless, an elongation factor, and different subcellular markers marking membranes or nuclei in these cases. So you can use this to generate ratiometric imaging in situ inside these plants. And so with these kind of approaches, you can use the now available genome information to start mining the genome itself for candidate sequences, which will be of interest in these rapid assembly tools. This is a brief summary of the Marcantia genome, where we have 8 autosomes and a X or Y sex chromosome system. There's something like 19,000 genes, and you can see the chromosomes themselves run from 20 to 30 megabase pairs. And the average number of, or the average size of each gene is spaced by 10 kb, and they're relatively compact in terms of number of genes, but the genes themselves are spread out. So one of the main features of interest here, which probably ranks as well with the simplicity of the experimental work, one of the main features is that Mercantia, as a liverwort, has a very simple regulatory circuitry. So this is from a review by Dolf Oges and Doris Wagner, showing Mercantia here with the key genes, of course, involved in auxin response, looking at tier 1 auxin binding factors, the AuxIA proteins and the ARF proteins, which are present at 27 and 23 and 6 in Arabidopsis in number. In Mercantia, there's a single tier A protein, there's a single AuxIA protein, and one of each of the 3 classes ABC of the ARF proteins. So it has these highly reduced numbers of genes involved in auxin response, and that appears to be the case for many of the other regulatory circuits that one can see inside Mercantia. So it has this great appeal that it lacks a lot of the redundancy which masks function in higher plants, even model plants like Arabidopsis. So using the fact that there's less than 400 transcription factors in Mercantia, we've made a comprehensive collection of proximal promoter domains from the transcription factors as a challenge, or as a way of mining particular interesting patterns that can be reused in engineering experiments. So basically to make a toolbox. And this is work that's been going on for a number of years now, most recently taken over by Facundo Romani and David Anisa, plowing through the screening of this library. So here we've got a series of transcription factor promoter elements showing the kinds of patterns of expression they drive. They're each here driving expression of a yellow fluorescent protein around the meristem, more broadly throughout the gemma outside the meristem, through rhizoid precursors and through oil cells, through the body of the gemma. So this ability to try and to build specific promoters allows us to start thinking about the next phase of things, which is how do gene circuits connect with cellular dynamics? And that, of course, is a major issue. And here we've got multicolor targeted cell types looking at their behavior during growth in the first few days of growth, just to illustrate the fact that these are live markers and directly accessible through growth. So using these to analyze cellular dynamics, Mihails-Delmans has done some interesting experiments which illustrate the benefit of this kind of approach. So for example, taking one of these gemma, you can visualize the growth, and you can see in this box, this orange box, that there's no cell division taking place. These are cells that are expanding. All the division is taking place around these meristematic zones at the top and bottom of these domains. However, in this case, Mihails has taken a razor blade or a scalpel and separated the meristem domain from the central domain. And so when this happens, you see this great proliferation of cell divisions that take place. So the meristem still divides, it still expands somewhat, but the central portion of the gemma, which would normally not divide, is suddenly de-repressed and starts dividing very rapidly. And so he documented this looking at, you can see here just in a period of a few hours, you see the onset of these rapid phase of cell divisions in these central portion of the gemma if they're separated from the meristematic zones. And so we now have a clue as to what's going on there. So you remember that we've got this cell division that kicks off if you separate the meristem. However, if you use a synthetic auxin present inside the systems, you see that you suppress this cell division. What's more, you also see the differentiation of rhizoids emerging from the central zone, triggered by the auxin application. You'll also see that the meristem ceases its rapid division and also starts expanding in a way that's similar to what happens in the central portion of the meristem. So using these kind of approaches, we have identified interactions across the tissues which are involved in controlling the meristematic and cell division properties of the tissues. And so using single cell transcriptomic analysis, Marius Rebman and Mihails-Delmans have together created a map of patterns of gene expression inside these tissues, where the dividing cells are shown up here, rhizoids are down here, and various photosynthetic tissues have been deconvolved from these transcriptomic studies. And these are all using 4-day-old GEMMA. So using this, what Marius has done is to start to look at pathways, for example, the auxin response pathway, and then map these different cell types and look at the preponderance of transcription in these different cell types. So in these, for example, you can see that there's the formation, or the genes that are associated with the production of auxin are generally found associated with the meristems, and those genes that are associated with the response to auxin, shown down here, are generally found in those cells that are in the central portion of the meristem. Sorry, my thing's gone wild here. So using these observations, he's also looked at cytokinin response, seeing where many of the genes associated with cytokinin response are present in this layer of cells, which is adjacent to the meristem, but not part of the meristem. So we're building these models for interaction, the production of auxin at the meristem, the flux of auxin to the central portion of the meristem, and the coordinate play of auxin and cytokinin in these transition zones around the meristem, using these mapping techniques to try and deduce interactions, and then using this information to as a basis for reprogramming cells to start to manipulate these dynamic processes inside cells. So it's a challenge to bring together patterns of gene expression and gene interactions together with the microanatomy of these tissues, enabled by the fact that mitochondria is so accessible and can be used directly. So this, in a way, brings together that diagrammatic view of that challenge, using these simple tissues as a starting point or a target, and having these increasingly useful DNA toolkits as a way of manipulating and perturbing existing regulatory networks and the interactions with the sort of downstream worker genes, if you like, and looking at the higher level of the way individual cells are interacting, which of course is one of the key issues in modern plant developmental biology, and I think also is extremely important for the new types of breeding approaches. We're already seeing that with the work of companies like Inari and Zach Libman's lab, looking at interactions between domains within meristems and probing and perturbing those in specific ways. And I think the use of model systems where we can directly visualize gene expression and cellular interactions is clearly critical for taking this forward. So I just want to finish with really thanks to all the people involved in the work, both past and present. And I think that for me anyway, this talk is maybe slightly strange in the sense that we've tried to marry this engineering approach, looking at electronics and programming, together with what I think is going to be the future of plant developmental reprogramming, which will embrace more and more of these challenges of using more formal engineering processes to deal with complexity in these systems and move towards more predictive and predictable ways of manipulating plant systems. And thank you very much for your attention.
Thank you very much, Jim. So we may have time for one quick question. Someone? I think everybody is hungry.
Yes, lunch.
Lunchtime. Okay, so, well, yeah, it doesn't seems that we have questions. So thank you very much for being with us today. And it was a very nice talk. Thank you.
Great, thank you very much.
So people, we now go into the lunchtime, and at 2:15 we'll be back for Flash Talk session 7. So please be here, and at 2:45 we'll be with our last keynote conference, given by Dr. Paul Getz. So see you soon. Thank you, Jim. Okay, so we are starting into the next session, Flash Talks. So go ahead, please. Flash Talks. So here we are.
Hi, my name is Coral Martinez. I'm a PhD student at the Institute of Biotechnology UNAM. I'm going to talk to you about my work and what results we have until now. The title of my work is Tisular and Subcellular Localization of a Protein Induced by Water Deficit in Arabidopsis thaliana, the case of HLA-45 protein. And to introduce you to the project, I will talk to you about what is a LEA protein. They are called late embryogenesis abundant proteins, which accumulate during the last stage of seed development, where they were discovered originally, and in vegetative tissues in plants under water deficit. They are ubiquitous in the plants. Most of these proteins are hydrophilic and disordered, and they can get structured under water deficit conditions. There have been studies for our lab trying to uncover the relevance of group 4 LEA proteins in plant tolerance to water deficit in Arabidopsis thaliana. This group is formed by 3 members, HLA-41, 2, and 5. Mutant plants in any of these members are more susceptible to water deficit. Also, in vitro assays demonstrate that HLA-42 and HLA-45 proteins could protect activity or reporter enzymes when they were subjected to partial dehydration or freezing defrosting cycles. This evidence indicates that these proteins could be exerting a protective function in the plant against the water deficit. Nevertheless, little is known about if their function in the plant is limited to certain tissues and cellular compartments. In my project, we want to explore the localization of HLA-45 protein, since it's one of the most studied members of group 4. Using fusion to GFP at the N-terminal of C-terminal of the protein. So far we know that HLA-45 protein is localized in imbibied cysts and some days after germination. And at least this protein localized in all the tissues of imbibied cysts. In seedlings exposed to soil, this protein changes its localization to vascular tissues from roots. At subcellular level, this protein is localized in nucleus and cytoplasm. Until now, we know that the localization of HLA-45 is different depending on the development stage and condition, at least at tisular level, which could suggest us the different process in which this protein could participate according to the development stage when the plant is subjected to a water deficit. Finally, our project's perspective are that we want to determine to which specific organelles or structure correspond the cytoplasmic localization of HLA-45 in embryos, and if this localization changes in another development stage and conditions. Also, we want to extend the analysis in the future to the other 2 members of the group 4 in order to know if all of them share a similar localization or not. Thank you so much for your attention.
Hello everyone, my name is Gustavo and I'm going to talk about amino acid signaling and the regulation of Arabidopsis thaliana root growth. Amino acids have been critical in the origin and evolution of organisms acting as structural units of proteins, signal molecules, and precursors of secondary metabolites. In plants, they also serve as nitrogen sources, as precursors of phytohormones, and as signals that induce defense systems and regulate stomatal movement. Besides, a previous report showed that L-glutamate affects the primary root growth of Arabidopsis when it is applied in the root apex. In the present work, we investigate whether, in addition to L-glutamate, other amino acids impact primary root growth and whether they converge in the same molecular mechanism. After comparing the effect of the 20 canonical L-amino acids on the root growth of Arabidopsis seedlings, we found that 50 of them were biactive. However, the most potent amino acids were L-leucine, L-lysine, and L-tryptophan, as they decreased 50% or more of the primary root length from 100 micromolar. The next question to be considered was, what are the targets of these amino acids to alter the root growth? So employing the transgenic transcyclin B1 gas, which helps to mark the cells in mitosis, we found that these biomolecules reduce the meiotic activity in the root meristem. Furthermore, they also shorten the size of the meristematic zone and the elongation zone. Then we observed that the NPK6 mutant affected in a gene encoding a kinase was less sensitive to the amino acid treatment than COL0. Finally, through an in-gel kinase proof, we detected that the different amino acids increase the NPK6 activity. So this result suggests that NPK6 mediates the amino acid signaling to diminish the primary root growth. You can consult all this information in the following paper published this year by the Plant Science Journal. Thank you for your attention.
Hi, my name is Genesis, and now I'm going to talk about differential expression of Arabidopsis glycine-rich domain protein, ATGRDP2 gene, under abiotic stress and hormone application. Abiotic stresses are responsible for almost 50% of crop losses worldwide. Cold stress affects principally tropical and subtropical plants like maize, rice, and soy. Recently, glycine-rich proteins were described to be implicated on abiotic stress. ATGRDP2 gene encodes an unknown function glycine-rich domain protein that it was implicated on growth and salt stress response. Herein, we characterize ATGRDP2 transcriptional regulation under cold, salt stress, and abscisic acid application. This was performed through a transcriptional fusion of ATGRDP2 promoter and GOS gene reporter. On pro-GOS thin line seedlings, we observe an increase in GOS signal in root tips and zone elongation under cold stress, in contrast to control and salt stress treatment. However, abscisic acid triggered the highest GOS expression in both temperatures. Besides that, we analyzed two more concentrations of ABA for a long period of time, and we observed at 24 hours the both treatment induced the promoter. Nevertheless, the high expression was in the secondary roots of 5 micromolar of ABA. On the contrary, at 48 hours, the highest signal of GOS was registered in the treatment of 0.5 micromolar ABA. Finally, at 72 hours, the high expression of GOS was in the secondary roots of the 5 micromolar treatment. Then, at 4 degrees temperature, we observed that in both treatments, GOS signal was very similar, except for the 5 micromolar ABA treatment at 72 hours, which had the highest GOS expression at this time. Finally, we analyzed salt stress on this line for a long period of time, and we observed at 72 hours of treatment, both types of roots had the highest GOS signal. However, when we subject them through a both cold and salt stress, the GOS signal decreased. And this is something really interesting because the root deformation on the salt treatment is not visible on the 2 stress treatment. All these results suggest that ATGRDP2 gene responds to abiotic stress and abscisic acid. This is all. Thanks.
Hello, everyone. I'm Ricardo, an inner student in the Immunogenetics and Allergy Research team, including Dr. Montero, Dr. Porras, Sweat, and Dr. Tran. Today, I will speak to you on a project titled Allergenicity Profiling of Ligostrum lucidum pollen IgE binding proteins associated with its geographical location in Mexico City. In the West, there has been a growing incidence of this disease in recent years. It is estimated that today, the prevalence of allergic disease worldwide ranges between 30–40%. Allergies have significant impacts on the quality of life of those who suffer from it, and especially in Mexico City, the prevalence is 42.6%. And there are many allergens in Mexico City, and one of the main one is pollen, which causes disease such as rhinitis or asthma. The most significant amount of pollen found in the city is caused by the Planaceae and Olaceae families. This graphic shows that Fraxinus excelsior is the most frequent pollen among the tree pollen. Fraxinus is a member of the Olaceae family and shares some allergenic proteins like OLE1-like with other members of the Olaceae family. For example, Ligostrum lucidum is commonly used as ornamental bushes around the world. In Mexico, it has an extensive distribution, so it is a risk for sensitized patients. But these families have the possibility of provoking cross-reaction in allergic patients. Other factors such as UV light and high CO2 concentration affects pollen development to be more resistant. The impact of the nutritional environment on pollen quality is not known. With all this information behind, we ask, is there chemical and morphological variability in Ligostrum lucidum pollen from Mexico City related to its allergenicity? To answer this question, we evaluate the proteomic variability of Ligostrum lucidum, testing serum from patients monosensitive to pollen, found in different locations in CDMX, and how these microenvironments can influence the allergenicity. Once collected pollen was sieved, we extracted the protein following these steps: precipitation, protein washing, protein hydration, and its quantification. With the estimates of the Bradford curve, we ran a polyacrylamide yield protein standard to observe how good the extraction was. Here I show you some of the most representative yields. Using image J, we analyze the yields to arrive at normalization of the protein load and proceed to do a slot plot to test the allergenicity of each sample based on the ROI obtained from image J and the amount of pollen protein obtained. We constructed this clustering to see which samples resemble more to whom. There's still some experiments we want to try, like the cross-reaction with Fraxinus. We suggest that the most stressed environments give the pollen more significant protein variability and consequently greater allergenicity. The most relevant regions, such as the center, are those that had the greatest variability.
My name is Aribel Sili Gutierrez. I am going to present the following work titled An Arabidopsis Transformation Plan for Sensitive and Tolerant Ecotypes to Test Biotechnological Strategies and to Improve Submergent Tolerant in Plants. The authors: Santiago Mayra Flores, Alexis Ortiz, Eric Barrera, Blanca Estela, and Peña Julián Mario. Submersion is an important plant product constraint, but a few cultivars of superior tolerance or ability. Arabidopsis is well established as a model plant that can use lab results to field applications. We aim to characterize a transformation platform with different ecotypes that can be used in the future development of transgenic submergent tolerant plants. We select ecotypes previously reported by Batish et al. 2011 to find the ecotypes tolerant and sensitive. The major ecotype tolerant is C24 and the less tolerant is CBA. The quantification leaves damage around 6 days in submersion stress, the ecotypes sensitive and ecotypes tolerant. The best strain of Agrobacterium transforming frequency, the ecotypes in Arabidopsis thaliana, was GB3101. The ecotypes C24 and CBO were the most contrasting ecotypes regarding submergent tolerance can be defined using an individual leaf damage score and can be effectively transformed by the GB3101 strain of Agrobacterium. Next, and the second part in this work, the study MIR1560 expression inhibitor flowering in juvenile plant state with the plant for biotechnological constructed plasmid with hemoglobin plant scavenger oxidonitric and BCIP sucrose homeostatic regulators with MIR156 recognition site 3' UTR to increase genetic transformation with age. Thanks for your attention.
Hello everyone, I would like to talk about the characterization of this TCEUS1 gene during mutualistic interactions. The process of nullation consists in the recognition of null factor by the plant cell, allowing the formation of the infection thread. The entry of the rhizobium triggers a series of responses like rose burst and cell wall degradation. The perception of cell wall-related molecules by receptor-like kinases like CRRLK activate modifying enzymes that allow the adjustment of cell wall properties. One of these receptors is TCEUS1 that has been described as a negative regulator of the cell growth and a sensor of the cell wall. TCEUS1 consists in two mallectin domain, one transmembrane domain, and one kinase domain that is important in the activation of NADPH oxidases. First, 1 kilobase transcription and fusion of TCEUS1 gene promoter was performed with GUS-GFP. The promoter activity was performed using a histochemical approach and RT-qPCR for determination of TCEUS1 transcript accumulation. An analysis of disease regulatory element present in 1 kilobase upstream of transcription initiation, we found 2.1 GM and 2.2 GM boxes related with genes activated during nullation process. TCEUS1 activity analysis showed external activity in root hairs with infection threads, and activity was observed during the first dividing cells that formed the nodule primordium until the formation of the mature nodule. The levels of TCEUS transcript accumulation correlate with the results obtained during the histochemical approach, showing a greater accumulation of the transcript in nullated plants compared with non-nullated plants, with the highest accumulation point at 14 days after inoculation with rhizobium trapezium. Considering the results, we can conclude that TCEUS1 has 6 regulatory elements related to nullation. TCEUS1 gene increases its expression during nodule development, and promoter activity of TCEUS1 is highly active during infection thread formation and mature nodule. Thank you for your attention.
I am going to present the following work title: A structure of substrate replica about Generación Bar Italicum. My research as the Master of Science Eduardo Márquez Ortega and Chemical Engineer Nadia Joaquín González González. The reaction better to know as broccoli has with benefits such antioxidant, vitamin, minerals, selenium, and glucosinate, from which sulfurafan derives reactive compounds with antimicrobial, anti-cancer, and antioxidant properties, but this property is lost the broccoli cookie. Glucosinate added derived from mustard oil.
currently from cauliflower, cabbage, and broccoli. The latter contains the hexamone research, and Johns Hopkins University in France has positively demonstrated in mammalian studies the eradication of Helicobacter pylori deep sulfurafane. The following images show how glucosinase are transformed with hydrolysis into sulforaphane from the mechanical damage that exclusively tends to sulfur cis component. Is hydrophobic, thermosensitive, and very reactive, so is pH to neutralize the ammonia and relax pH pylori inside the gastric mucosa. It might show how sulforaphane acts inside the stomach to protect antimicrobial barrier and guide conscious this component is good. Catalyst of the NRF is due then when activated relax anti-cancer cells and isofol type 1 to make a cellular ready to a food chemotherapy. Of this part of the process is research and development for the formulation of the antibiotic routine of several circulation was carried out where the hydrolysis process took place from the sulforaphane. The non-solar irradiation of day was done before a solution of citric was added to the sample to avoid sedation. The sample was checked to show the right texture to later extract or with chocolate technique. You observe the vapor and decontain to obtain the sulforaphane. The following image shows part of the rewriting fluorescence and plumboard showing the right texture to extract the oil. Theoretical data of sulforaphane contain influence and plum flakes and will be used to for content-specific consistency experiments. Also, we was carried on a range of 16 for 34 years to find out 1% of the population segment to suffer with gastric ulcers and stomach cancer, as we had hoped they could now avoid its property and hope the smile is the gap is consumed quickly. It seems loose as its property included in the active compound. In conclusion, the research based in a theoretical manner indicates the significant of the process contained of the licenses and testing and quite consider important points, which are to magnify their few ways, design and stage methodology, to create new products using sulforaphane in the pharmaceutical and chemical industry.
Hi, I am Cristóbal Hernández from the Faculty of Science of the UNAM, and I will talk you about my PhD project in which we explored bacterial diversity from arid zones with the capacity to interact with plant roots. It is thought that bacteria inhabiting dry localities are adapted to those conditions, so plant-interacting bacteria from these localities could improve crop resistance to drought. With this in mind, we collected samples from arid and humid localities to characterize the bacterial diversity. Additionally, we conducted a greenhouse experiment under controlled conditions using the soils to grow squash plants. At the end of the experiment, the plants were collected and their phenotype was recorded. The microbiome analysis by 16S ribosomal RNA gene sequencing revealed differences between samples from arid and humid localities. However, it was remarkable that bacterial communities from the local samples and the common garden experiment were not separated, suggesting that local diversity was successfully transplanted to the greenhouse. As we expected, sterilized controls were different from the rest of the samples. Generally speaking, Communities from arid localities have a major proportion of actinobacteria in the soils, the root surrounding area, and inside the roots. Some taxa were enriched in the root samples from the arid locations. From that subset of bacteria, some genera were correlated with plant phenotype. For example, the relative abundance of Salvibrio was positively correlated with aerial biomass, foliar surface, and the quantity of leaf pigments. Finally, metagenomic analysis indicate that arid communities are enriched in genes for stress sensing, maintaining osmotic pressure, and avoiding oxidative stress. But also, several genes may be involved in the interaction with plants and other bacteria. We conclude that bacterial communities from arid sites were different from those of the humid ones and rely on mechanisms to face drought. Some differential taxa from arid sites were correlated with plant phenotype, indicating that these bacterial communities could improve plant survival during drought conditions. For more detailed information, you can read the complete paper here. And if you're interested in the plant microbe interactions, you can follow our lab's Twitter and find more related papers in our ResearchGate profiles. Thank you very much.
Hi everyone, I'm Mara Cucinotta, a researcher in the Plant Development Group at the University of Milan in Italy. Here I'm showing you my project regarding the study of vacuolar protein sorting 13 during female germline establishment in Arabidopsis thaliana. In our group, we focused the attention on the study of ovule development, and in particular, we focus on the process of megasporogenesis. So, in the process that bring to the identification of a single megaspore model cells and the commitment of these cells into meiosis. As you can see from the picture, knockout mutant of VPS13 is affected in all aspects of plant development. The plant is very small with several problems and in the end is completely female sterile. We decide to investigate more in the detail the problem in ovule development. In order to avoid the pleiotropic effect of lack of VPS13, we decide to specifically silence this gene only in the ovule using the SPL promoter. And in this PSPL VPS13 silencing line, we have 30% of ovule abortion. Here you can see that the marker for MMC identity, the knuckle YFP, was switched off in the majority of VPS13 mutant ovules. And in the silencing line, also the problem of on the identification of a single MMC are visible. Moreover, the silencing of VPS13 lead to aberrant callus pattern during meiosis, and the majority of the ovule in the silencing line does not show callus staining. Later, by using a marker for the identity of the functional megaspore, we observe a very particular phenotype in the VPS13 silencing line. Indeed, one of the spore does not correctly degenerate and acquire also a functional megaspore identity, leading to the formation of more than one functional megaspore. At the moment, we are currently investigating a possible link between VPS13 and the small RNA pathway. Since VPS13 is co-expressed with many genes involved in this pathway, and mutant of genes involved in small RNA biogenesis or silencing through macroRNA show phenotypically defect during ovule development very similar to that of VPS13 mutant, such as the block of outer integument group. So thank you for your attention. Don't hesitate to contact me for any further question. Bye.
Hi everyone, I'm Carla Esquivel and I am a graduate biotechnology student from Universidad de Papalapan. And the project I'm working with is titled Design of Double-Stranded RNA-Based Biofungicides for the Control of Plant Pathogens Rhizopus oryzae, Fusarium incarnatum, and Geotrichum candidum. Important export tropical fruits like pineapple, melon, banana, and papaya are highly produced in the tropical humid and subhumid region of Papalapan in Oaxaca, Mexico, where diseases caused by fungi affect crop production at any stage of the food chain. In a previous work, fungi before mentioned were isolated and identified as the principal pathogens of the region's tropical fruits, and in this work is pretended to evaluate the use of spray-induced gene silencing technology for its protection. SIX technology is a modern biotechnology strategy consisting in the use of double-stranded RNA molecules that target against specific genes, silencing its function. And for this, dicer-like gene sequences were identified for each fungi, whether they were available in databases or predicted by using different bioinformatic tools. And once the step was covered, specific primers that amplify regions of 250 to 500 base pairs in length were designed by choosing low similarity regions with their respective host fruit DCL genes. Once DCL genes were amplified by PCR performance and fused, which is the case of the two DCL genes predicted for Fusarium, T7 promoter sequences were added to both ends and then ligated to a vector and cloned into competent E. coli cells for subsequent sequencing for sequence confirmation. The perspective of these works is to synthesize long and short double-stranded RNA molecules that are later going to be used for activity bioassays, which consist in the evaluation of spore germination activity in vitro according to Mombanza methodology and fungi real growth inhibition on PDA medium, to finally collect all the data that is gonna to be evaluated by statistical analysis. And that's the stage of my project so far. Thank you so much for your attention.
Okay, so we continue with our last keynote conference of the Congress. So I really appreciate, and I'm honored to introduce to you to Dr. Paul Getz. He is from the University of California, Davis. He will tell us about molecular and environmental aspects of domesticated Middle American common bean. Thank you, Paul, for accepting our invitation. The audience is yours, so go ahead.
Can you hear me?
I think you're muting, or
No,
try it again.
Can you hear me?
A little bit, it's a little bit lower.
Can you see my slides?
Yeah, I can see your. Maybe it's the volume. You just have to.
Can you hear me?
Yes, but it's still is very
It's pretty strange.
low.
I tested it before,
Now, now I think it's better.
Yeah, okay. So I'll start and.
Yeah, it's now okay.
Okay, so I would like to thank you, Alejandra, for the invitation, and also the organizing committee for inviting me. And I'd rather be in Mexico, but frankly,
sure.
we all know what this is. So my program is really focused on trying to understand the genetic diversity of crops, and for the purpose of their conservation, but also utilization in crop improvement programs. And the approach I've taken is to establish kind of a phylogenetic framework, as well as an environmental framework, to understand this genetic diversity. And my favorite experimental system has been the genus Phaseolus, for reasons that you will understand later. And it has applications to bean improvement. And the example I will illustrate today is drought and heat stress tolerance. So in my talk today, I will speak at the beginning, kind of summarize and present the experimental system, Phaseolus, the genus Phaseolus, which is a domestication hotspot, especially in Mexico. And then in the latter part of the talk, I will then document the search for tolerance to heat and drought in this the genus. Now, the general framework of this research, it turns around domestication. And as I said, Mexico is a domestication hotspot for the genus. And this process of domestication has three types of variables. It has the characteristics of the plant itself. It has also then environmental conditions under which this domestication take place. And thirdly, and not the least, is the human factor, which is really the driving agent of domestication. So in this talk today, I will talk mostly about characteristics of the plant and the environments. Lack of time, I won't be able to talk as much about the human contribution, but it's definitely the case. We have data, genetic data, showing the effect of selection of farmers, both in Latin America as well as in Africa. And farmers do exert selection, and this selection has a measurable effect on the genetic diversity of their stocks, the seed stocks in their respective farms. One of the characteristics of Phaseolus beans is that their main role is as role in the human diet. And so you see here a series of bean dishes I've collected around the world. It's mostly a dry bean, but it's also a green bean. And even in some part of Mexico, they eat the flowers, and in other parts of the world, they also eat the leaves. Now, this is not just kind of pleasant to look at, but it definitely exerts a selection effect on the genome. For example, on the colors of the seeds, their size, their shape, their texture, and so on, their cooking time, and so on. And I think that it's up to now, we have done very little to try to map and understand at the molecular level what these selection effects do on the genome. But it is important, and so in Mexico, Phaseolus beans are an important crop. I think that it's the second or third crop in Mexico. So I mentioned before that Mexico is a Phaseolus domestication hotspot. The genus has some 70 to 80 wild species, and 5 of them have been domesticated, and 2 of them have been domesticated actually twice, once in Mexico and a second time somewhere in the Andes. And you see here a photo here put together by
postdoc, Travis Parker, who shows the 7 domestications and their respective wild ancestors here. And so the 2 species that were domesticated twice are common bean and lima bean. And I'll come back to that later on, to the right part of the photo where you see tepary bean and lima bean. And the questions, well, some of the questions are, what are the genetic and molecular basis for these differences in adaptation among these 5 species? What is the effect of domestication? Which genes have been selected? And it looks like there is some type of a domestication potential, but we have very limited understanding of that concept so far. The species show, the genus itself is a fairly young genus, and about 4 to 5 million years. And against this common genetic background, we see a tremendous differentiation for life history, for adaptation, for reproductive system. So it's, in a sense, a very interesting model that we have this common genetic background and yet tremendous differences. And so later on, I'll talk about tepary bean, lima bean, in addition to common bean here as a control. So standard research compares the Andean and the Mesoamerican common bean. You can then compare them to their respective wild ancestors. But you can also compare the 5 domesticated species and their respective wild ancestors. One of the things that we have tried to do is to understand the genes that condition the domestication syndrome. So a syndrome is a series of traits that are characteristic of the domesticated condition. There seem to be a certain repetition among very different crops in this syndrome. One of them is the change in the growth habit. You have generally a more compact, less branched growth habit in the domesticated types and less competitive. You also have a reduction in the photoperiod or sensitivity, and beans tend to become insensitive to the photoperiod as they move from tropical or subtropical area to temperate areas. You have then a change in the seed type. You see at the left here the seeds of wild Phaseolus vulgaris, and here a sample of the domesticated diversity of those. Common bean has been selected as a green bean, and you see then also the variability appearing in terms of color, the pod shape, and so on among the pods. The wild relatives have dormant seeds, and that dormancy has been lifted in order to have regular germination. And then an important trait I will talk more about is the loss of pod shattering. Wild beans shatter their pods at maturity, and domesticated beans do not, or much less, much lower extent. Now, over the last decade, my group and several other groups in the Phaseolus community have isolated some of the genes involved. And you see here, Koinange repeated here among these different genes. This is the first experiment we conducted to map these genes, and their location has been verified now several years after that. And I'm particularly pleased that we were right the first time. And these genes have been isolated in the location we predicted in that first paper almost 20 25 years ago. Now, what do these genes tell us? You see here the plant function. So there are different functions here. The particular trait, the gene on which the chromosome on which the gene was isolated, the type, and that's really important. We see there are 8 genes of the domestication syndrome that have been isolated. It's about an equal proportion of transcription factors, of structural genes, and of genes with that code for enzymatic function. All of them, all 8 of them, represent loss of function, and this is consistent with their recessive nature, and I'll come back to that later on. So how do you achieve loss of function? Well, it's the usual catalog of all ways you can inactivate a gene. For example, the determinacy gene, the major way in which this was achieved by a retrotransposon insertion in the 4th exon of what is a homologue of the TFL1 gene of Arabidopsis. But there are other mutations as well. You see deletions in part of the gene, or non-nucleotide substitutions. And so you see really, like I said, a catalog of different ways of achieving a loss of function. Now, I would like then, having introduced kind of the experimental system, most of the research has been conducted on common bean. It's really economically the most important, but also geographically the most widespread of these domesticated phaseolus species. And a recurring constraint on the production is drought and or heat tolerance. It has been estimated that about 60% of the area where beans are grown is subject to drought stress. And you see examples here on the left where you see areas here in Mexico that are either heat and or drought stressed. But the same holds, for example, in the second largest, most important area or continent that produces beans. You see that also in Africa. And in Mexico, it's a recurring problem year after year. There is, to more or less extent, there is drought stress. Okay. So I'll illustrate this with 4 topics, and I'll talk first about the domesticated Phaseolus vulgaris. Pouch shattering is not only a domestication trait, but it's also a trait adapting beans to arid conditions. Next, I will ask the question whether the wild Phaseolus vulgaris, the ancestor in Mexico, can contribute factors that that allow beans to tolerate drought, and will go as far as introgressing wild beans into a domesticated background and see if that can work. I will ask the question if sink strength, whatever that concept is part of a mechanism of stress tolerance to drought. And then finally, I will move out of common bean and into other domesticated species, and particularly what is called peppery bean or Phaseolus acutiformis. So the first topic then is this pouch shattering. So this is a wild bean growing in, as it happens, Mexico. And it's at maturity, the plant is dry and the pods are dry also, and after a while the pods shatter. So what does that mean? You see here a close-up, and you see that the pods, the 2 valves of the pods separate, and each valve twists strongly. And this is an explosive movement, and so it propels the seeds in a radius of about kind of 5 meters around the plant. That mechanism has been lost, not completely, but to a large extent, in domesticated beans. And so you see here, and I showed this, you see the twisting is still present, so this is a problem variety. It does show still pod shattering, and this is a negative yield factor. And so the paradox is that on one hand, there has been selection against this pod shattering, but on the other hand, we still see this atavistic trait, this ancestral trait reappearing as part of the breeding program. So why is that? So we started looking for the molecular basis of pod shattering, and first in the biparental population, we have 2 varieties here, and the shattering was characterized in 3 ways. A very simple way, it's in the field, just saying or looking presence or absence of pod shattering. The second way was to take a sample of pods from the field into the lab and in a oven expose the pods to high temperature for about a week, and then count the proportion of shattered pods. And then a third way is here is the force measure, which is to measure the force that is needed to have the pod open, actually. And you see that the 3 measures actually identify the same genomic location on chromosome 3, but with an increasing degree of confidence. You see the lot scores increase from 8.9 to above 50. So we are pretty sure that this is the location in this population of a gene. It turns out that under the area of high significance, there is a pod dehiscence gene. It also synthetic to soybean, and this gene is what is called a dirigent gene, so it participates in the synthesis of lignin. It adds individual lignin units to the larger polymeric lignin molecules. This is a second example. Here we looked at a panel, diversity panel, both a Mesoamerican and an Andean diversity panel, reflecting the 2 domestications of common bean to see is the same gene involved, yes or no. You see that the tip here is that PVPDH1 gene, but you see other factors on other chromosomes that are also significant. In the Andean domestication, the major factor is actually a mid-transcription factor on chromosome 5. So a different location, a different gene involved. The one here is not necessary. Not sure that it's the same gene as in the Mesoamerican domestication. Okay. Now, an additional observation we made is that in the Mesoamerican gene pool, this PVPDH, the allele that reduces shattering or eliminates shattering, is not distributed at random. It is concentrated in the varieties that grow in the northern part of the country. And this is what is called race Durango. It is a race that is primarily distributed in the highlands under dry environment. And you see here, the orange dots are that low shattering allele, and they are concentrated pretty much in this area and in this group of varieties. On the other hand, in race Mesoamerica, which is a lowland ecogeographic race, much more humid, there you see a much higher level of pod shattering. And so the idea here is that following domestication, what we think is central Mexico, probably in Jalisco and the western part of Guanajuato, that following the domestication, there was a migration of beings towards the north, forming raised Durango, and selection of a loss of function mutant, the PVPDH1, to allow it to produce fully and avoid the pod shattering. On the other hand, there was a migration towards the lowlands and creating a race Mesoamerica. And because the environment is much less dry, there was not a systematic selection for this absence of pod shattering, because the humid environment does not induce the pod shattering as severely. So this mutation actually is in the PVPDH gene is also not only a kind of domestication trait, but it's also a factor of tolerance to arid conditions. And this slide then summarizes the work of many of our lab, but also others, and also the homologies with factors in other species. And you see that it's fairly widespread. It's not only legumes, we have Arabidopsis involved, and even a cereal. But they are distributed over all these chromosomes. And that explains then, in part, why we still see varieties today. So this is kind of a phylogeny of the species, Phaseolus vulgaris, or common bean. There is a lineage that started several hundred of thousands of years ago that eventually gave Phaseolus vulgaris as we know it today, as it has been taxonomically classified. But this lineage spread out. There was a first long-distance movement to Ecuador and northern Peru. These wild populations have since then relabeled or renamed into a Phaseolus debuckii, because they show some distinctive traits, not so much morphologically, but metabolomically, as well as in their distribution, which is unusual. And then this took place about 400,000 years ago, and then another migration took place 100,000 years ago and gave rise to these wild populations in the southern Andes, which were domesticated, giving rise to land races, and then eventually to domesticated varieties outside the center of origin, and all these improved varieties. And the same holds then here. Eventually, this lineage gave rise to a domestication here of land races in Mesoamerica, and then further evolution under breeding and dispersal from the Americas. And so we have these two domestications, and we need, where do these mutations that reduce pod shadowing, where do they fall? And you see here that they are distributed in both domesticated gene pools and different ecogeographic race. And we have seen this with other traits as well, is that you have these gene pool or lineage-specific mutations. In the case, I think the multiple mutations here seem to suggest that farmers have paid particular attention to this problem of pod shattering. And we see the same thing, for example, with the determinate growth habit, where you have one gene has multiple mutations. So they're definitely an important trait to consider. Now, why is it then that we still, even though this is a domesticated trait that has been selected against, why do we still see shattering? And you see here different market classes that still show high level of pod shattering. You see, for example, the cranberry kidney beans here. You see also the small blacks, like you have in Mexico, and then the navies. And my contention is that there are two causes to this persistence of an atavistic trait. One of them is that it's the redundancy in the genetic control, and all of them are recessives, so they complement each other when there is a cross between the two. And then it's the environmental influence on the expression of the trait. The trait shattering is only expressed under the driest of areas. So both the combination of both explains why the trait is not eliminated systematically from the domesticated gene pool. But it also illustrates that these domestication traits are not just interesting for the past, for what happened during domestication. There are still traits that are important for the productivity of current varieties. I will now ask the question, the second topic is, can wild beans actually be a source of higher yield and drought tolerance? In domesticated beans, the motivation for this is that it's a well-known phenomenon, is that there is a reduction in diversity during domestication. And I've listed here a few examples. There is a sharp decrease in molecular diversity, and also wild beans have been a source of agronomic traits in the past. We have not tested so far the potential role in drought tolerance. Now, there is a difficulty in evaluating wild beans for drought tolerance. It's difficult to evaluate yield because they are climbing beans, because they tend to shatter. There's a different selection regime between the wild and domesticated types, and that is reflected, for example, in the much higher harvest index in the domesticated types. They exhibit also a stronger population structure and a narrower adaptation. So evaluating wild beans for drought tolerance is really difficult, doesn't really make sense. One way, an alternative way to do it is to cross wild beans to a domesticated tester and look at the progeny. Now, this is a considerable amount of work, and we propose that one way to pare down the number of wild population to test is to concentrate, to select the potential wild bean sources on the basis of their relevant environmental adaptation. And you look here at the left, the distribution of wild beans, which go from northern Mexico to northwest Argentina. It's about 10,000 kilometers. There is a high number of populations, but these populations come from fairly different environments. There is always a dry period, but around the tropics, it's a short period of 1 to 2 months, and at the extreme of the distribution, the dry period can reach 6 to 7 months. And so if we go, northern Mexico has a very, as you probably certainly know, is a very dry area, and it's combined with a high temperature, so a high evapotranspiration. So this leaves us with a possibility of testing this, and what we did was to actually take 3 populations of wild beans along a gradient of humidity, of rainfall, one from Durango with 500 millimeters rain, all the way to Guatemala, just south of the border, about 2,000 millimeters, and then cross these to a same parent, C5, which is a breeding line with tolerance to drought, and cross them, have 1 back cross, and then 4 generations of selfing. And during that generation advance, we selected against photoperiod sensitivity. And so you see here the frequency of the different lines in terms of flowering time. And on the right here, we eliminated those lines because they were photoperiod sensitive and would not complete their cycle under the conditions of Davis. And you see here at the bottom here an example of the breeding nursery. To the left are other plots of the breeding program unrelated to this, but they represent domesticated by domesticated crosses, whereas the experiment here you see is a green square reflecting the fact that these are later materials in spite of the selection against photoperiod sensitivity. Nevertheless, we were able then to measure the yield of these materials and at the same time genotype the lines with a SNP chip, and you see the QTLs that were identified. I'm not going to go into this, but you can actually map the QTLs. Under well-watered conditions, we identified 9 QTLs. However, the effect they had actually limited effect in erasing the yield of the domesticated parents. You see here the additive effect, but also the percentage difference, and it's very small and it's negative, but very much it has no effect under well-watered conditions. Now, if we examine the yields under drought, the picture was quite different because the populations that came from the driest or dry areas, the two populations, increased in a statistically significant way the yield of the domesticated parents. In contrast, the population that came from the wettest area had also no effect. So our hypothesis that we can select a subset of these wild populations based on what we think is their adaptation, in this case drought, seems to work. And we can consider wild beans as a potential source of drought. Now, why is it? What are the traits that actually in these wild populations confer this drought tolerance? We conducted an experiment in the greenhouse in these long tubes that are about 1.20 meters long, so as not to limit the growth of roots. And we can then do what is called a network analysis that calculates the correlations between the different traits or different variables we consider. You see here the result of this network analysis. At the top, you see the input variables. You have environmental adaptation of the parents, not in the experiment itself, but in the area of origin. And so it's temperature, rainfall, aridity, and so on. And you also have the treatment. You have drought and well irrigated. So these are the input variables. All the others here in the central part are the traits, the phenotypic traits we measured. And at the bottom, you see here the ultimate response variables. And these are the variables that distinguish wild beans from domesticated beans, and also wild beans under conditions or well-watered conditions, and drought conditions. And so there are two types of traits. We have the root trait and the growth vigor. So wild beans, when they are stressed, they will modify the shape of their root system to concentrate on a deeper root. That it confirmed also that these wild populations from arid areas have deeper roots in general. But when you impose a drought stress, the wild beans will remodel the shape of the root system to concentrate on a taproot that reaches more deeply into the soil. The second trait is that growth vigor. If you impose a drought stress, wild beans tend to continue growing, whereas domesticated beans tend to slow down in their growth. So these are two traits that we should concentrate on when we try to look at what are the specific genes that we should identify and transfer into domesticated types. Briefly now, as I get towards the end of my talk, I'm asking the question, is sink strength important for yield, and especially under drought stress? The picture you see here is kind of a bean breeder's dream. You see these pods and they are filled completely with one seed after the other. What we see is that under stress, you start seeing a loss of seeds. You see gaps in these pods, and we have been able to verify that in a growth chamber, in growth chambers, to see that, for example, heat stress or drought stress imposes a symptom of that is the loss of seeds within the pod. Now, the question is, we did an experiment then where we imposed a drought stress and then measured what is called the post-pod harvest index, which is the ratio of seed weight over the weight of the total pod, so pod walls and seeds. And the results are that we identified 9 QTLs for pod harvest index, and a major one responsible about 30% of the phenotypic variation on chromosome 7. Yield, we identified 8 QTLs, and 3 of which were clustered or were the same as the pod harvest index QTLs. That suggests that an important component of the yield buildup is that pod harvest index, the ability to translocate photosynthates from the pod walls into the seeds. Okay? And I should also mention that as part of this paper, we saw substantial epistasis. And that is usually not considered, but we usually consider only the additive effect, but the epistatic effect also play an important role here. So overall, this experiment underscores the importance of photosynthate remobilization. And the question is, how do we study this further, and how do we select for an efficient translocation, partitioning, and remobilization, all the way from the leaf into the stem, then back into the inflorescence, the pods, walls, and then into the seed. The pod harvest index is only kind of the last step of that translocation process. Okay? So we need additional criteria to select this thing. I think one possibility is here is the so-called suspensor. This is research that is conducted in Bob Goldberg's lab at UCLA. They have published these 2 papers recently. So the suspensor is a structure that is at the basis of the embryo. So you have a suspensor here, and then at the top here, you have that, what they call the embryo proper. So this is a very young embryo. You can dissect it out of the pods, and they like to use Phaseolus coccineus because it has such large seeds and large embryos. And you can then, what they do also is to do laser capture microdissection and able to then isolate the messages from the suspensor and the embryo proper. Okay? And what they see is that primarily the messages in the suspensor are for hormone factory and the transfer of substances, and I quote them here, to the developing embryo. And my question is, what are those substances in addition to the hormones? Are they photosynthates, for example? Whereas the embryo proper has primarily transcription factors, cell division, and genes involved in development. Okay? So clearly there is a difference there, and the question is, what is the expression under stress? I don't know, but I think that it would be a valuable direction of research. In general, as I mentioned before, this is the bean plant and other plants are made of modules, and these modules are stacked. This paper here, Tanaka and Fujita, demonstrate that most of the photosynthesis stays within the module. So again, to repeat my question is, how do we document efficient and high level of translocation from the leaves into, for example, the inflorescences at the axil of the leaves? That is a question that is, I think, unresolved, but that would help developing more productive crops, including also the effect of stress. And so lastly, I would like to look at other species. I've mentioned that we have 5 domesticated species, but there are 2 species that grow under environments that are drier and or hotter, and these are tepary bean and lima bean. And lima bean was also the other species that was domesticated twice. So this is an experiment that is, or research that is ongoing. It's a collaboration between CIAT in Colombia, University of Nebraska, and UC Davis, and it provides a comparison of tepary bean and lima bean with common bean. And you see the locations here with their respective climates. And the result here is that if you compare, for example, these 3 species at UC Davis, which is a relatively cooler environment, and the West Side Research and Extension Center in the San Joaquin Valley, you see systematically that tepary bean, whether it's under well-irrigated or drought conditions, comes out on top. Lima bean is next, and then common bean, under certain circumstances, barely yields anything. So this is not a candidate for a global warming scenario. Definitely we ought to look at these other species. On the right side of the screen, you see the variation within tepary bean. Not every tepary bean variety is tolerant to heat and drought. Certain land races are, and so you should have screening. And then the TEP lines here are breeding lines, and they show the benefit of selection, the immediate benefit almost of selection. Okay. And so there are alternatives, and I think that these species, to the extent that we may have to switch crops in certain regions, that these, because they are related to the currently preferred crop, common bean, because they are related, they are better candidates for substitution. So, in summary, then, I would like to mention that I showed that tolerance to pouch shattering is an aridity adaptation trait and certainly a yield factor in an environment that will become drier as we speak. Wild beans can provide tolerance to drought under specific conditions. So the choice of wild populations, the method of introgression, and the type of trait that we look at. Pod harvest index is a useful parameter to assess yield under drought stress, but I ask for role of the suspensor in that scheme, and also we should be able to assess each photosynthate translocation step from the cells of the leaves into the seed, and we don't have these tools right now. And then finally, there is a lot of diversity in the genus, including its domesticated species. They provide a potential solution for a drier environment. So thank you very much for your attention.
Thank you so much for this insightful talk, Paul. It was really interesting. So just because it's your last talk, maybe we can have one question. No one? Well, I may have one question. So when you're talking about drought, you mean drought at what stage of the plant?
Yeah. So we have considered two types of drought. And what I presented today was primarily terminal drought.
Okay, good.
The type of course, that favors tepuridine, which escapes drought through a short cycle and as well as other features. I think limabine is more tolerant probably to intermittent drought, where you have, rain, drought, and so on, because it has a very deep root system that can. So these are the two main scenarios, I think, for drought that we consider.
Exactly. So you haven't looked at more earlier drought conditions, such as during very early stage of the plants?
No, the experiment in the greenhouse,
mm-hmm.
that was early growth before flowering.
I see. Okay.
Yeah. Here, so this was from germination all to the V3 stage, which is trifoliate stage, but before flowering.
Just before flowering. Okay. Yeah. So in the mature stage, yeah? Right?
In this case, not a mature stage.
Yeah. Okay. So thank you so much for your talk. It was really, really good, and so we'll go into the next part of the Congress, and I hope to see you soon.
Yeah, I hope so too.
We should talk, actually.
Yeah, I think you would have an interest in the translocation, I believe.
Yeah, exactly.
Yeah.
Okay.
So thanks for inviting me. It was a great pleasure.
Thanks to you, Paul. Nice to see you. So, so we're going into the very last part of our meeting, and so for the next part will be the session where we will tell you about the awards. So I'm going to share my screening. And if you don't know, but for the ones that don't know, in the last meeting, it was established a prize for posters and talks of the students, and this award was entitled in honor of Dr. Federico Sanchez Rodriguez, who was a very important part of our plant community, a very enthusiastic researcher and a very good friend of mine, at least. So this time we decided to, instead of giving one prize, we are going to give various prizes because we have a very good flash talks. And so the amount that we decided to assign for each prize is here. And I will tell you about the prizes we get. First, of course, I have to give a special thanks to our judges, Gustavo Rodriguez, Elizabeth Cordova, Alexis Acosta, Pablo Peláez, Paul Rosas, Héctor Torres, Moraina Gutiérrez, y Lidia Cabrera. To them, very, very much thanks for your collaboration. And so these are the names of the different prizes we are given for the different sessions of the flash talks. So we are given 2 first prizes, 3 second prizes, and 3 third prizes. So Jara Servín and Fonseca will be first for the Monday's session, and Mezquita and Aguilar Cruz and Ortiz Martinez second, Granados Aguilar, Pulido Torres and Garcia Coronado thirds. And for Tuesday, Maldonado Maria and Lara Mondragon first, Gomez Hernandez and Cervantes Bueno, and Gomez Diaz second, and third, Solis Miranda, Buendia Monreal, Deep Alvarez. And for the Wednesday session, the first awards were for Petrella, Fonseca Garcia. The second prizes were for Huerta Perez, Rodriguez Cisneros, Ortiz Luébano, Cázares o Casares Álvarez, Rosas Reinhold, y Pacheco Cruz. And I'm sorry, but for these last flash talks, we still don't have the last list, so we'll let you know who are the ones that got the awards for these last flash talks. And so for all of them, congratulations and thank you for participating in this meeting. All the flash talks were very, very good, and I think we had a very good information and good data from these talks. So with this, I think we go into the final part of the Congress. That would be the concluding remarks that Dr. Felipe Cruz will give. So Felipe.
Yeah, thank you, Ale.
Yeah.
Could you share your screen, please?
I'm gonna leave my screen, just wait a minute, okay?
Dan, could you share the screen, please? Hello, Dan.
Who's gonna?
Dan is going to put there.
Okay.
Yeah, the problem is that Dan is not a co-host right now, so please, Edgar and Cedric, make Dan a co-host. Yeah.
Hi, Dan. Thank you, Dan. Just to mention some of the statistics of our Congress, I think that it was very successful one because you can see there the numbers. We had 1,300 participants, 36 speakers, 107 flash talks, 50 countries participating, and more than 3,000 YouTube views. And there is something that is very, very important, that the age of the people that was attending this Congress was between 25 to 34 years old. This is very, very nice because this is the future for the plant biology area. Also, I want to thank the organizing committee, Alejandra, Mario, Ulises, Stefan, and Dan, and the technical team from the School of Chemistry of the National University of Mexico, Sonia, Edgar, Cedric, Adolfo, and the team that was working in formatting the abstract, Dulce, Olivia, Liliana, Adolfo from the Universidad Veracruzana, and the acknowledged is by their deep commitment to making this Congress possible in a very successful manner, as shown by the large numbers of participants and its significant reach to different continents. I also want to thank the speakers who did not hesitate to accept and give us their beautiful lectures, leaving many topics to reflect on in our minds. Also, I want to highlight the great work with the very high quality that the students and postdocs presented in their flash talks. It makes it clear that you can say a lot and be fascinating in a short time. I want to thank the Mexican Society of Biochemistry and the American Society of Plant Biologists for their valuable support for the realization of this Congress. Finally, as demonstrated in this pandemic, I would like to highlight the resilience that human beings show to achieve what we set out to do when we work together for a common goal. Thank you very much. Muchas gracias a todos. Y sí se pudo. Thank you. I pass the voice to Stewart, that he is going to be the next president of the organizing committee for the next Congress in 2023. Go ahead, Stewart.
Okay, thanks. Thank you very much, Felipe. And thanks to the current committee members and the participants of the meeting for having the confidence in me to select me to be the president for the next meeting. It's an honor, and I'll do my best. We're working on finalizing the complete committee that's going to be involved, but it's looking like it's going to be a great committee. So I'm really excited about that. As Felipe said, the plan is to have the next meeting 2 years from now. If anyone has any ideas or suggestions for topics, speakers, locations, or anything else, feel free to send me an email. You can find my email address on the Lankebio website. And I'd also like to mention that it would be for the participants here, if you're not a member of the Sociedad Mexicana de Bioquímica, it would be great if you sign up. And if you're not a member of the Mexican section of the American Society for Plant Biology, it would be really great if you sign up. It only costs $5 per year, 110 pesitos. And really, it's the Mexican section of the ASPB has only been going now for a couple of years. It's fantastic that we finally have one. I was hoping we would have one for like 25 years, and now we have one. And it's a really great way to let the world know about the science that's going on in Mexico. The ASPB is one of the oldest and most important scientific societies in the world, and it's the American Society, and Mexico is in fact part of North America, even though many people don't want it. So it would be great if you sign up. You can go to the ASPB website or ask Patricia Leon or Stefan de Folter how you can sign up. I think that's about it for me. You'll be hearing from us from the new committee relatively soon. And again, if you have any ideas, feel free to send me an email. And thanks again to the committee for selecting me as the president.
Thanks, Stuart. I don't know if somebody else from the committee wants to say something. Ulises, Alejandra, Dan, Stefan, anyone that wants to say something, go ahead. No one?
Maybe you can switch on all the camera, the committee. Just saying thank you to all the participants. It was really a great Congress. I think we all enjoyed it ourselves as well. And maybe a bit nervous before the beginning, but it turned out great. Thank you for participating. And thank you, Stuart. We are looking for a nice in-person meeting in 2023. Thank you.
Indeed, in person.
I just want to say that thank you to all the registered people and the flash talks that were contributed, because we were not expecting that such a big participation, but we are really happy that everyone put a lot of effort on the flash talks, and also thanks to the speakers who, from the very first moment, they said yes, I want to participate, and it was really good because these are difficult times, and we have to be up to the moment. So thanks a lot to everyone, and I'm looking forward to the meeting in 2023.
That's it. It's done.
Just one final word. I want to thank everybody, but also I want to bring to your attention that at the very beginning of organizing this, we were not sure what was going on, what was happening in the world, and what was going to be the situation. And I remember Felipe called me, and we had this very existential talk about how to go with the meeting. And at the end, I think it was really the Felipe was the driving force behind putting everybody in contact, and his enthusiasm for doing organizing the meeting. I think it was something really important, because this was organized like 1.5 years ago. We started with this, and the situation in the world was really troublesome. So I just want to give a special thank to Felipe, because thanks to him, I think we all came together to try to do a good job. So thank you, Felipe. And thank you all, everybody, and all the team and everybody backstage. They did a great job. Thank you.
Yeah, I think we should thank Edgar and Cedric mainly, because they did a very great job helping us with the technical issues and all the things. So to the technical team, thank you very much. And also thanks all, and see you the next meeting, right? Bye.
We did a very great team. Dan, do you want to say something, Dan? What is your impression to all the Mexican people?
I have nothing more to add, but I just remember when we were at the last meeting together in Mérida in 2019. It was just before all of this pandemic started. I don't think any of us could have imagined that when we were in Mérida, which was so nice and great, that this could have ever happened. And yes, just to echo what everybody else said, I don't think any of us had really imagined a virtual meeting like this coming together so nicely. I think there's other virtual meetings, but we made something, I think, a little bit unprecedented. It was a completely free meeting. There was no registration at all. Anybody on the internet could watch the meeting. There was so much student and postdoc involvement. And I think we made a lot of positive changes to the way we go about our normal science, even in very bad times, through this meeting. And all of us discussed that we hope that there's some great location in 2023 in Mexico that we're all together again, Oaxaca, I don't know, someplace nice. But this hybrid aspect will be great, that if somehow we can make the meeting accessible through these new technologies that we all learned.
I'll take that as the first suggestion for a locale for the next meeting.
It is the official first suggestion.
Okay.
Okay. So thank you all.
That's it, right?
See you.
Thank you, everybody.
Thank you.
Bye.
Thank you. Bye.
Thanks. Bye.
Bye.